Good morning, everybody. Please take your seats and we get started. I hope you all had a nice, relaxing evening and got to enjoy the beautiful weather we're having up here. I spent half of it walking my dogs. It was wonderful. So we had a great day one. I was really inspired yesterday from all the sharing of knowledge and all the stories we heard, and we're going to carry it on with day two. And today we are going to hear from a number of the researchers who've been working on the questions that were essentially inspired by the last Beluga Summit. And again, the presentations you're going to be seeing here are the sort of presentations and research that the community members, Game Council, FJMC, have asked. But beyond that, there is, as you can tell on the walls, there is a plethora of other work that's really exciting and that also provides greater context for the state of knowledge on beluga whales. So today I'm happy to present our chair, Jason Stowe from DFO, and he'll be helping guide you guys through today. All right? Thank you. Jason.
Thank you, Jen, and thank you to the organizers for giving me this honor of chairing the session today. My name is Jason Stowe. I'm a manager with the Arctic Ecosystem Science Division at DFO. I'm based in Winnipeg. I've been there since 2018. Prior to that, I worked at the Northern Contaminants Program Secretariat since about 2002, and it was in that position where I became familiar with the fantastic work that happens here in the Inuvialuit settlement region, and specifically the beluga monitoring that takes place at Henriksen Island. So going on, pretty close to 22 years that I've admired the work coming out of this region. Am I close enough? Yeah, okay. All right, thanks. So as Jen mentioned yesterday and touched on a bit today, the Beluga Summit 2 guided a lot of the research and monitoring the Beluga Summit 1 guided a lot of the research priorities that have been addressed over the last 8 years, including, from Beluga Summit 1, we were told we needed to do an accurate population assessment on the eastern Beaufort Sea beluga. And you're going to hear about that today. A health assessment, how are the beluga doing? How are they reacting to changes going on? What the health of beluga means for the health of Inuvialuit. Where do beluga go when they're not, when we can't see them? How do they move around? And then finally, a priority was for community engagement and the mobilization of Inuvialuit knowledge along with the science. And so you will see in the agenda today that each of these topics are going to be addressed in topical sessions. We're going to start with beluga and country food health, and we're going to hear 2 talks plus an introduction from the leaders of that session, Lisa Lassetto and Jim Elias. Then we'll move into a presentation from Sonia Ostertag and Richard Grubin on country foods for good health. Each of these talks will be followed by an opportunity for questions and answers. And then before lunch, we're going to talk about some of the lightning talks. So what you're going to notice in the presentations today is they're a fairly high-level overview of what represents a tremendous amount of work. Where you will find a lot of the details is going to be in the posters that are around the room. And I really encourage you to take some time looking at those posters and speaking to the presenters of the posters. Some have multiple out there, because I think that's where you might find some of the details that you're specifically interested in that aren't necessarily covered in the short amount of time we have for presentations. After lunch, we're going to get into a session on distribution, abundance, and habitat use with presentations. This session is going to be led by Manny and and Shannon. And we're going to have presentations from Marianne Marcoux, Luke Story, Lisa Lassetto, and Emily Couture. We'll then break, and there'll be an opportunity to look at posters, and then we're going to have some breakout sessions, which we'll introduce at that time. So without any further delay, I'd like to ask Jim and Lisa to come up, and they're going to give you a little bit of an overview of the first session.
Thank you, Jason. Thanks, everyone. I just wanted to open up and highlight with this slide here that's also depicted in a poster highlighting a lot of the different questions we've been asking with the harvested whales. A lot of the things that you see listed here are being supported by research and monitoring programs that have been identified through community concerns, meetings, community observations, and knowledge. What we've done here is build, I heard someone call this yesterday, one of the most studied beluga populations, and that's not because, it's a population that's not doing well, it's a very healthy population, but it's because we have such a strong collaboration and the Nunavut leadership in wanting to know what's going on and being proactive and doing things like setting up MPAs, but also through setting up MPAs, we are put to task to make sure we're monitoring what's going on. So I want to acknowledge all of the work is a true collaboration. And then last point, yesterday I heard someone talk about beluga as being the canary in the coal mine, and that's very much true in that the beluga is telling us about ocean health, and it's giving us information, and we're here and we're listening, and we're happy to share what we're seeing and what the beluga is telling us through all of these studies.
Yeah, thank you guys. I'm Jim Elias too, and I work with Lisa quite a bit, or be involved with Lisa's work quite a bit in that, but just want to highlight that, how we're working together now. Like back in the day, we used to see researchers come into our communities and that, and we don't even know who they are and what they're doing in our community and that, but now that we're working together, more involvement with the researchers, they get to know the people, and then when they even do move on, their person that's taken over for their spots, they're already involved with the community, and you know the people. And like the pictures there, back in the day, the black and white pictures, we didn't have to worry about any contaminants or stuff. Everything was just beautiful, pristine world we lived in back then. But now you see what's happening all around the world, where there's so much different kind of diseases and everything that's happening in that. So like Lisa said, we want to be proactive in that. We want to keep our population healthy. That's our traditional foods, and we want to know what we're eating when we do harvest the animals and that. And saying that, they're almost like us, where we pass on our tradition to our children, and they pass that on to their grandchildren and stuff like that. And then when the researchers like Lisa and Sonia and them come up here and that, they've been up here so long, even get to know their children also and stuff like that. So it's like kind of like a community involvement now. Just want to bring that up. Yeah, and saying that though, that's just my little blurb there, but I gotta put my glasses on to call up some people here. So Emily Couture will be first up on our list here, and then we might also call up Adam Morris and Amila De Silva. Come up. I'll turn the floor back over to Lisa.
All right. Good morning, everyone. Thank you for being there bright and early this morning with me to talk about beluga health and foodborne parasite assessment and the TNMPA. So my name is Emily Couture, but I do want to acknowledge all of the collaborators for this project that actually started when I was still a veterinary. I was a veterinarian, but I was pursuing a specialization and working with wildlife, and this was a very important project, I guess, in my professional pathway, and I'm very thankful to have been part of it for almost 10 years now. So what is the health assessment? So first of all, it's a project that's been supported by the IGC and the TUC HTC. And the health assessment presented today would not be possible without the structure that is already in place for monitoring beluga health and doing research, which is led by DFO, the FGMC, and with the participation of the community hunters. For the health assessment, what it consists is we are a team of veterinarians and veterinary technicians that come onto Hendriksen Island and join the already present team, and with a little bit more support for DFO because we come with a lot of work and tasks to do. And we basically look at beluga health. And we also partner with the University of Saskatchewan to look more specifically at some parasites that could be transmitted through food and assess their presence in belugas. The main goals of our projects are informed by the priorities that have been determined. What we want to do is we better want to understand nutritional condition in these wells. We want to be able to evaluate change in beluga health through time by taking snapshots every 5 to 10 years, basically. We also want to, as I mentioned before, evaluate the presence of foodborne zoonotic parasites. Is this something we should be worried about or not? And we want to be able to monitor the reproductive status in female, which can then help inform stock assessments. And then with our expertise in anatomy, we can support sample collection that can help a lot of other projects as well. Small example of that, we were there this summer and we were able to provide, different organs to try and come up with cell lines to better study toxicology in belugas. So far we've had 2 health assessments. Each health assessment covers 2 harvesting seasons. So we started in 2015-2016, where we sampled 26 adults. And we just completed the field portion of the second health assessment last year with a total of 35 adults sampled over 2 summer seasons. So this is great. We have a total of 61 wells included in the information that I'm going to present to you today. I'm mostly going to focus on what we find in the field. Part of this, but not just, but part of this is because we're still looking at all of these samples that we collected. And if you want more details on what the health assessment comprises, you can go and see Amelia and her poster with all of the steps, including everything that we do once we're back in the lab. So first of all, for the evaluation of nutritional condition, the first thing, and one of our first goals when we first came on the island, was to develop a reference indicator, something that would be ironclad in helping us assess the body condition of the whale. And that needs to, what we need for that actually is have a whale weight, which is more easily said than done on an island in the Arctic. So this has been a journey. So wearing belugas in a field setting such as Hendriksen has, to our knowledge, not been done before. The first health assessment was a testing ground with some humbling beginnings. We had a very bulky mobile setup. It was difficult to properly raise the whales above the water. We needed to bring the setup over to the whale every time we wanted to weigh one. And overall, it ended up in unacceptable delays for the hunters' time and collaboration. Even with that collaboration, I mean, we got a few well-deserved, doubtful looks on this setup. I think if people were on the island in 2015, we can all remember and cherish the memories of my mentor, Stefan, trying to crank that lever hoist with all of the oil he had in his elbows to try and do it as fast as possible, but not getting the results we really wanted. We did manage to weigh 10 whales, and we're very thankful for the participation of the hunters for that. But after that, and we had a good 7 years to simmer a little bit on this, and we came back in the second health assessment with an upgraded system. So basically, what we've been doing in the last 2 seasons is we add a skidder designed especially for belugas to be able to drag the belugas out of the water using a portable winch, which Jasmine is here operating, and to then bring them under a better tripod and lift them off the ground. Of course, the first few times are always a little bit longer, but again, patience and participation from the hunters was really well appreciated. And we have here on the right, so a group of future leaders that were part in helping us do a proper setup to lift wells. And with this new setup, we've been able to weigh 23 wells, so a majority of the wells that were sampled in that second health assessment. The main change that this brought is that it's now a stationary weighing place. So the harvesters come to us to weigh the wells, and it has worked really well. It made collecting some samples, such as the blood, which is pictured here, easier. We've had amazing collaboration, and so one of the things that we started doing is giving out T-shirts with the weight of the wells to thank harvesters for their time. What the weighing setup helped is to help take better pictures of the shape of the well so that we can ultimately build a catalog and help have a better and more precise visual assessment of their body condition. Based on that visual assessment of body condition, generally I can say that wells appear to be in a good body condition, and the rest of our analysis is to try and dig a little bit further into that. And I think you will all agree with me that these pictures, it's much easier to look at the shape of beluga from this than when we try and take the pictures and we haven't quite been able to take them out of the water. The other indicators that we take to try and look at the nutritional condition of the wells, a lot of them have been monitored for years by the program in place. What we've added is a few of other measurements based on the work we do with the St. Lawrence belugas, for example. So we're taking a lot of half-curse, the total length of the well. We're taking blubber depth. We also take samples, and one of the side projects from this program is to look on the microscope at the shape and size of the fat cells themselves. And one of our hopes is to try and find a region of the blubber, maybe the portion that's more towards the animal, that would be more sensitive in being able to tell us about the body condition of the well. Because there are tick specialists, there's probably a good part of the blubber layer that doesn't change due to body condition because they need this insulation from their outside environment, basically. So we're going to try and dig a little bit deeper into that. And then the other thing that we've been looking at is muscle, because there are tick specialists, there might be a point where if they need to go and get energy, they might be getting it from their muscle before their fat, because they need their fat as insulation. And this is one looking at the weight of the muscle in St. Lawrence belugas was a better indicator sometimes for body condition. So we're going to see if that could also be helpful in the Beaufort Sea belugas. The ultimate goals with this, so we come in, we take a whole lot of information, right? But the goal is not to always be doing this. We want to take these indicators and put them against this reference indicators that we're going to develop to try and find which ones, maybe they're the ones that are already being monitored, but maybe there's new ones that can be incorporated so that you can help inform the monitoring programs. Then there's the health indices. So health indices in belugas, and again I'm looking at beluga health. Belugas, they live and they're exposed to a harsh environment. They will encounter harsh conditions. They can get injured, but they're wild animals. They should be resilient. They do form strong communities, and they live to an old age. We might be seeing changes in their body that are reflective of their age. And as any wild animal out there, they are naturally exposed to parasites or other microorganisms, but the chances are that they are in a balance with these. So they are what we call an host, and then there can be, so here are certain pathogen. You can just think of anything that we touch on surfaces, microbes or parasites. But there should be, in a healthy situation, there should be a balance, and they should not have a negative impact on the well. So this is what I want to look at. whatever we find, does it have a negative impact on the well, and is this something that is changing through time? In the next few slides, you are going to see pictures of parasites, and I know that, I mean, I see it from a veterinary perspective where I know that it's from what we've seen so far, it's in a very healthy balance for the whales, and I know that they do not represent a risk for consumption for humans. So I do want to stress this out, because I know that it can be, when you're a hunter, to see some of the pictures that I will be showing can be sometimes, and we've had these discussions on the islands before, but just keep that in mind. The whales are healthy. The parasites that we're seeing are not causing any issue, and they're not a concern for human consumption. And with this in mind, maybe you'll get as excited as I am when you see them. So what I'm mostly going to present is the examination that we do with our naked eye. So what do we see when we open the well? And we don't just open the well, we take all of the organs out and we slice them up to try and see if there's anything within the tissues. One of the first thing that we see, and that's pretty much in all of the whales, is those little circular depressions on the skin. So we've looked at that on the microscope from the first health assessment, and it looks like it's just a regular molting process. The fact that we see some of them on the back of the whale, especially at the front, like we can see on the top picture, makes us think that it's potentially, and it's been written in some articles too, that it might be linked with ice breaking. So like, bumping against the ice, and maybe they injure a little bit, and that skin, when we eventually, when the whales get harvested, we can see those little circles. So nothing to worry about that. Now we're getting into the parasite part. So the kidney worms. So there's, as you can see here on the figure, this is where the kidneys are. The kidneys, there are a bunch of little masses, right? Each of this little ball is a small kidney in itself, and this parasite manifests itself as a white lump that we can see in the kidney, but it only takes so small of a portion of a kidney, it doesn't affect the whale health. This parasite that we call crassicauda is known in some other stocks of beluga, and I'll circle back to this. Sarcocyst. So sarcocyst, that's a parasite that we cannot see with the naked eye. It is found in the muscle. It's also a family of parasite that I can say that we find in a lot of game species, including in Canada. And they're not, even though we don't really like to think about parasites in our food, they are not known to infect humans, and it is not a concern for us. Another interesting finding that we've had through the health assessment is these little white lumps in the lymph nodes that drain the gut of the animal. So these, when we started looking at the microscope after the first health assessment, what we saw is that we could find the eggs of a parasite, the type of parasite that we call flukes, in there. I'm focusing on these three parasites first because these are all new observations in East Beaufort Sea belugas that come through the health assessment. As I said, they have no impact on harvested whale health, even though the kidney worms have been seen in some other beluga stocks. It seems to be a fairly recent observations in more Arctic populations, those at the Chukchi and the Beaufort Sea. That's probably something that has been there maybe within the last 20 years or something. So things are changing, and it's good that we have already started this process so that we can keep monitoring this. What we have done with all of these three parasites is we've taken further steps to try and better get to know them. 5 minutes? This is, I might get a little bit over, but hopefully I'm going to answer in advance some of the questions you might have. I might speed up though. So basically what we've done, so we've partnered with Emily Jenkins' lab, who we're already looking at some parasites with. to try and further characterize the sarcosis that we're seeing in the muscle, and she's also interested in looking a little bit further for these nodules that we see in the lymph nodes. Same thing for crassicaula. So Rafaela, who's here, has been working on and is very interested in learning more about the ecology of these parasites. So these parasites, they basically help us maybe understand what is changing in the beluga environment, their diet, maybe where like their environment altogether, and then why we're gonna try and find out like why we are seeing this, and it didn't seem to be something before, basically. Another thing, and that's just very much expected in pretty every beluga population, we do find nematodes, so worms in the airways. We can also, when we look at the lungs, sometimes it can just look as firmer areas in the lungs. And we can see worms in the stomach. Sometimes it can be associated with ulcers. Most of the time it's just small numbers. And even if they are associated with ulcers, they are usually not a problem. The picture that I put on the right is one exception that we had, where maybe we can think that this animal maybe had a little bit of a stomach pain every now and then. But the worms themselves are not necessarily the cause of the ulcer. They might just take advantage of it to go and try and get a little bit more comfortable in there. So they are expected in small quantities, and they have no impact on whale health, and this is what we've been seeing in Beaufort sea belugas too. Last thing that we've seen is what we think are age-related conditions. We still have to look a little bit further into the samples that we took for this last health assessment. But so what we see is changes that are associated with the glands. So basically the thyroid, which is up here in the throat, and the adrenals over the kidney. And we generally see them in older animals. What we see is little masses and maybe like little bubbles. And this is probably part of the normal aging process, and it is to be expected. Regarding foodborne zoonotic parasite testing, so I want to talk about, Cody here has a poster where if you have any questions, you can go and ask more questions about the work that has been done about the Jenkin labs. I will not, I will cover it a little bit. But what I want to stress out is that there's no significant risks that have been identified so far. So Trichinella is one of the parasites that they've been looking for. So that's a parasite that basically in the life cycle of that parasite, it's within animals that eat other animals. It's mostly associated with bears or walruses and carnivore species, basically. And it hasn't been found in any of the beluga that have been tested. So it does not seem like, and it makes sense because they don't really go and eat red meat, right? So that's good news. And then the other parasite that has been looked at is Toxoplasma. So I think if you were part of the Beluga Summit back 10 years ago, this might have been a topic that has been addressed. Basically, the thing that I want to stress about the foodborne zoonotic parasite testing, yes, they partner with us with the health assessment, but the foodborne parasite testing, it goes on continuously. There's testing every year, not just when we're doing a beluga health assessment. So there's a lot more animals that have been looked at for this assessment than what I presented for my work. And basically it's been found in one heart out of 173 animals, and that was 10 years ago. Toxoplasma, what we sometimes forget, so the only type of animal that can spread Toxoplasma and multiply it, they're felids. So the feral cats, or just the domestic cats, and the wild cats, which are probably less dense than the feral cats. And then anything that drinks or eat something that has been contaminated can harbor the parasite. It is something that we have and that we are exposed to down in the south. I probably am exposed every time I don't watch a carrot enough from my garden because my neighbor's cat come and use my garden as a litter, basically. And we are mostly concerned about these parasites for people who are pregnant and get exposed to the parasite for the first time when they're pregnant, or people whose immune system is not strong enough. Other than that, usually it is not something that will make people sick. And the good news here is that it is not something that we see in beluga. So I probably am exposed to it on a more regular basis than for somebody who eat beluga. So this is the take-home message here. Then again, for the reproductive status monitoring, so that's one of our last point. So in total, there's 13 females that have been looked at in over the 2 health assessments. Of these, 54% were either pregnant or had really recently calved. The youngest to oldest, so we had females from 13 to 49 years old who were pregnant. And in the non-pregnant animals, we had 2 senescent or menopaused, if you want, animals that were 51 and 55 years old. And that's very interesting because what we found in the literature is that they thought that females stop reproducing after 40. But even with the few females that we've been able to look at, we can start to think that maybe we can push that bar to 50. And that has an impact when biologists look at stock assessment on like how long a female can actually reproduce for. And if you want more information on what we found in that aspect, you can go and talk to Amelia again with her poster. So take-home message here, the wells that we looked at, they're generally healthy wells. And of course, that's compatible with why they're selected to be harvested, right? You guys select the healthy wells. We are still ongoing the 2023 and 2024 evaluation. We're looking at the slides right now. And the observations that we've done so far have and will lead to collaborations, further research, and overall better understanding of beluga health, ecology, and stock assessments. What's next? So again, we're going to complete that second health assessment, hopefully before the end of the year. We're taking, so this summer is a break, no field season. We can look at our samples. And then, so hopefully, third health assessment within the next few years. And in the meantime, we're working with our collaborators to maybe think about, is there already some things that we want to include more into the regular program so that we can go and get more information on the females, for example? What you might ask is, what if you find something outside of the health assessment? As a harvester, you harvest a well and you have a question on, like, there's something that doesn't seem right with it. For well health, what you can do is you can take picture, share your knowledge of the health of the well with us, and take a sample, freeze it. We are working on creating a better and more efficient pathway to get the samples to us and then to get the results back to you. And then by getting more samples and more of your input on the wells, we can collectively learn more on well health, because some of the lesions or the abnormal things that we find, they might not just show up in the wells that happen to be there when we're there on the island, right? For human health, do rely on your harvester traditional knowledge. It's very important. You have been doing this for a long time. And for non-foodborne parasites, there is continued monitoring through the FGMC and the DFO sampling, and the risk is currently estimated to be non-existent to low. This is example of samples and pictures that were taken and were submitted to us this year. So this is the first time that I received samples from outside of the health assessment, and I'm very grateful for the efforts that were taken in getting these samples to us. And this is something that we would not have been able to document otherwise. So this is very weird. On the left, there's very weird looking blubber. On a well that was harvested on Hendrickson, and the harvesters decided not to consume the well because it was a foul smell with the blood. What we saw on the pieces that we received in look on histology is basically dead fat cells, and there was no sign of infection or inflammation. Of course, it's always difficult to assess of the health of the general well or make sure that we find the cause or something. There's always limits to lab analysis. But right now, I guess the working theory is that maybe there was an injury to a vessel, and so blood stopped going to part of the blubber, and this brown discoloration that we see is blubber that died, and that's probably recently. That's probably happened a few hours to maximum a few days before the well was harvested. Did it have an impact that sort of like created maybe a blood clot? I can't say exactly what is the cause, but if we get more samples, maybe with time, and if more wells show up with that, then maybe we'll keep understanding more. On the right is a sample, so a first sample from East Whitefish. Again, thankful for the submission. This well is a very localized mass in the blubber, and it looks a little bit funky with all of these little nodules when we started cutting it in the lab. But again, what we've seen is that there was no infection associated, and it seemed like it was a very late stage of healing when we looked on the microscope. So I can't exactly say what caused it, but it was not an infection, and because it was so well defined compared to the rest of the well, it was very likely, and the harvesters in this case reported that the health seemed otherwise healthy. And I would strongly believe this, that it was just a piece that could be cut out, basically. Long-term dreams. With time and building up these knowledge on beluga health, we hope that we can eventually come with developing a tool that can help support harvesters. That could take the form of developing, and we've been talking about this for a while, and now we're starting to get enough data for it. It would be nice to be able to develop a guide using the gathered information and image so that hunters can have this type of information when they're out hunting. There's also a potential partnership with other scientists looking at other stocks so that we can combine our knowledge together. And then another long-term dream is to build local capacity to document and sample when you see something abnormal so that we can continue better inform outside of the health assessment, and then maybe also be able to respond if there is any stranding that happened that when we're not there, if a sick well, for example, washes up. So thank you very much. It's been an honor and a privilege for me to be there for a few sampling seasons. Thank you to our partners and funding partners, and to all of the people that stayed up late to do the health assessment.
Thank you very much, Emily. Fantastic presentation, beautiful photos, and it's easy to see the passion that you have for this work. Do we have any questions for Emily? We do have some time. Many?
Just looking at that, good morning. Just looking at the whale, good presentation, a lot of good information. That sick whale where is that harvested?
Hendrickson, the one with the brown piece of blubber?
Yeah.
So that's and that's great because it was the monitors. The monitors were still on the island, and they're the one who harvested the well too. And having been there through the health assessment, they contacted people from the FO. They took the pictures. They took the sample for us. It was good.
And all the organs and that too, they took samples of inside the stomach and that too, or
yes.
I just seen the outside.
So they think they took the samples, the regular monitoring samples, and we did receive liver and muscle for that whale too, to look at on the microscope, and we didn't see anything in these two type of tissues. From my side of things, it's hard to say without having looked at the whole well, so I trust their judgment on deciding not to eat that well.
And good presentation, a lot of good information. Thank you.
We have time for a couple more questions, if anyone wants. No? Okay then, so maybe what we'll do is... Oh, was there a
I
question?
see a hand raised.
Nigel, here you go.
It's sort of slightly random question, Emily, but the stomachs, are they often full or are they empty when they're harvested?
Good question. I have yet to see a stomach content in a beluga well, and the monitors can probably talk to that more because they've seen more wells than I. But yeah, Manny, did you have a? The only content I've seen so far are those little wormy things.
As a harvester, we always hear from our parents and our grandparents, and we go out hunting and that. Even I notice that they throw up their food when we start chasing, and that's what we always hear. And when we bring them back in, their stomachs are empty when we check them. And even you can ask the monitors. The beluga monitors that every time we check them that they're pretty well empty.
Thank you.
A question from Marianne.
Great presentation. You kind of hinted at the beginning of the talk that you are going to compare your results to other population. Do you have some insight?
Yeah, well, I guess I mentioned some of it. So if I go back a little here, I mean, some of the and this is part of what takes time, right? It's the comparison. It's compiling everything together. So we have done after the health assessment. There's been a global report done for the 2 first years, and that's already available, and it's even publicly available on the CWHC website. And so we've done that work for the first self-assessment. It took a lot of time. Now we're going to look, and I don't want to steal the thunder from Amelia's poster here, but once we've done looking at all of the slides and doing all of the analysis for the second health assessment, it's going to take some time to put all of the information we have together and also look a little bit deeper into the links, right? Do we see, so for the age-related lesions, like we're going to test that. Like, what are the ages of the animals where we see lesions or in the glands that we think are associated with age? And then so we're gonna, compare within our own results and with the other information that's getting that is getting developed by our partners. So age is because of our collaboration with DFO that we have access to this data. And then it's looking and comparing with information that is out there in the literature or directly with the people working with other stocks and trying to synthesize that together into a report that will now not just look at, it will look at the evolution through time from 2015, 2016 to 2020, 2024, and if there's anything new that came out for the other beluga stocks.
Thanks, Emily. One more question, Jimmy?
Good presentation. It's good to see passionate people working and collaborating with us, so very much appreciated. The question I had was on the beluga that had the bruise or the dead fat. I always had questions, comments, or concerns about killer whales.
Mm-hmm.
So we have the technology to see, you figure it might have been a couple of days that bruise happened within that range. So in the area, we have the technology to see if there's any ships in that area, or if it might have been a smaller boat to see if there was a strike.
Yeah.
If not, then I would like to look into see if it was a killer whale, because you know how killer whales they're like the wolves of the sea, and the way they hunt their prey is they're pretty smart animals, and they do like to
Bump into
hit
things.
them hard too,
Yeah.
and stuff like that. So
Yeah.
that might be another reason why that whale is injured that way.
Yeah, that's a very good point, and one of the possibility was maybe some sort of damage through some sort of impact to a vessel, and then death of some of the blubbers. So a ship strike if bumping, getting bumped by an orca, and by sometimes it's very anecdotal information, right? But if you record it once, then you might record it twice, and then something might happen. Somebody might see something happen, and then you can link your observation with, by gathering and by being careful and watching around. Eventually, maybe we might not have all of the answers, but with time and by gathering information, eventually we should get a clear picture.
Thanks a lot, Emily, and thanks for all the questions. If anyone does have another question or something comes to you afterwards, there will be opportunities to engage with the presenters at the breaks, and again, we have the poster sessions as well. One comment, Jim? Okay, but quick.
Yeah, so one comment there. I know Emily is very passionate to work and stuff like that, and then, especially when you see her working out in the field there, and then once you came into the building yesterday and that, I wouldn't recognize her without the blood and all the stuff on her and there. She gets right into it and that. So I just wanted to say thank you very much for your hard work.
Thanks, Jim. Okay, I'd now like to call up our next presenters. So we're going to hear from Adam Morris, Lisa Lassetto, and Emila Da Silva. And they're going to be talking about contaminants in Beluga and the monitoring program. Go ahead, Adam.
Stealing my thunder. Good morning, everybody. My name is Adam Morris. I just wanted to say thank you to the organizers for inviting me here. I've been working in the North for almost 20 years now. Makes me feel both a little old, but also incredibly proud of all the amazing friends I've made across the North in the work we've done together over the last decades to answer their questions about contaminants. So right now I work for the Northern Contaminants Program. I actually took Jason Stowe's old job, so he left me some pretty big shoes to fill. But I'm going to tell you a little bit about the NCP, the Northern Contaminants Program as a whole. So we are a federal science and funding organization. So for 34 years, we provided funding for the study of contaminants, also communications, capacity sharing, and knowledge exchange across the North. We have about $5 million a year that we allocate to different projects. This year, we funded 62 projects in total, including the long-term Beluga monitoring program. So we fund the contaminants portion of that work, as well as some of the communications work. And we also provided funding for the summit. Not a lot in the grand scheme of things, but we did provide some of the funding for that, as well as the first Beluga Summit as well. So really our mission, our mandate is to reduce, and where we can, eliminate contaminants in traditionally harvested foods. Unfortunately, eliminating contaminants entirely can be very difficult, or sometimes impossible, unfortunately. But our other main job is to provide the information on those contaminants to people in the communities so that they can make their own decisions about food use and what foods they want to eat. And we take that job very seriously. To achieve our mission, we work with Northerners and scientists, again, across the North on the research and monitoring of long-range contaminants and plastics. And I specify long range because a lot of the contaminants that we do study do not come from inside the North, they come from far away. And I will say, compared to down south around the Great Lakes, for example, the contaminants here are at quite low levels pretty much across the board. There are some other cases where things become a concern, but by and large, we're exposed in the south to many, many more contaminants than you are up here. The contaminants that do make it up here, though, have to get here somehow. So many of them do come through the air, or through the atmosphere, through different mechanisms, but that figure on the left there shows some of the ways that the contaminants might get here. Others do come through the oceans or through river currents as well. Those are the main ways that we get these contaminants introduced up here. Excuse me. And again, it's only a small portion of those that we see in the south that do make it all the way north, but some of those can be very long-lived and persistent in the environment and in animals as well. And when I say contaminants, what am I talking about? There's lots of different contaminants in the world. So we look at things, again, those that are likely to make it up here, or we know are up here. That includes things like heavy metals. Mercury is a big one that we hear about everywhere we go. There's also lead and cadmium as well. Then we have the organic pollutants. So again, these get a little more complicated. We call them POPs, but the old school legacy POPs include things like PCBs, which you may have heard of, or old pesticides like DDT. Once those were banned, we had to, industries created new versions of these chemicals. In a lot of cases, those also became a concern. So we call those new POPs. And those were also replacement insecticides, but also things like flame retardants and forever chemicals, which are PFAS chemicals, which you'll hear from Emil in just a little bit. We're also always looking for chemicals of emerging concern, we call them, things that we don't necessarily know are here, but scientists think maybe they might make it up here, or things people in communities have let us know that they're concerned about. So we're always trying to stay a little bit ahead of the curve and look for things that could be a concern in the future. And also more recently, we've been looking at plastic pollution as well, which has become a huge concern across the globe. We also look for little particles of plastics, but we also look for the chemicals that are in plastics that make them bendable or resistant to sunlight, and things like that. So, not going to go into too many details on all the contaminants, but yeah, there's lots to talk about there. Another important part of the work that the NCP does is with the Arctic Monitoring and Assessment Program, or AMAP. We've worked with AMAP for over 30 years. And they are an international group that brings together all of the Arctic countries to produce scientific assessments that provide a lot of information on contaminants and climate change as well. The work under AMAP provides a lot of information to the countries involved, but also to international treaties that work to regulate and potentially ban these harmful chemicals. So it's a very important organization. Again, they do a lot of important work and produce a lot of different assessments. If you go to the link on their website there, you'll find links to all these different assessments there as well. In Canada, and the Northern Contaminants Program in particular, has a lot of responsibility to AMAP because we do have a big chunk of the Arctic, as you all know. So, the long-term trends of contaminants and how they change over the years, like those that are generated by the Beluga Monitoring Program, have contributed to major international policies and treaties, leading to bans on harmful chemicals. So I think that's a really important point to note. But because these chemicals come from long distances, especially through the air, reducing contaminant levels in the Canadian Arctic requires a lot of international cooperation and agreements. Again, that's why AMAP is so important, but also these treaties that, again, the data from this program have contributed to so well. And those include the Stockholm Convention, which look at those persistent organic pollutants, but also the Minamata Convention on mercury. Mercury is a little bit different because we can't just eliminate it. It is a natural element. It will always be there in the background, but things like coal burning can be reduced, and in that way we can reduce the amount of mercury in the north. It's also really important to note that Indigenous peoples have always played an important role in these policies. The delegations are groups of people that go to the negotiations, always involve Indigenous representation. So the Arctic Indigenous Peoples Against POPs included the Inuit Circumpolar Council, Inuit Tepiret Kanatame, and a number of other organizations there. And so they were there, excuse me, at the beginning of the Stockholm Convention to make sure that it had things in it that established protections for the North as well. So all that to say that Arctic countries, when we stand in solidarity with our Indigenous peoples, can really impact global regulations and affect global change, which is a really cool part of our work. So I've talked a little bit about these long-term monitoring projects, and this is all the long-term monitoring projects that the NCP currently supports. I'm not going to talk about all of them, but it's marine mammals, fish, caribou, air, and seawater. And in the ISR, you can see that we have a Sax Harbor Seal Monitoring Program. We have the Long-Term Beluga Monitoring Program. And right here in Inuvik, we also have 2 air monitoring sites. And currently our caribou project leader, Mary Gamberg, is consulting with the Tuck HTC to get some caribou work done in the region as well. So lots going on in the ISR, and I just want to thank you all for all the hard work you put into supporting these programs, providing feedback to us, and to the researchers as well. It's crucial to the way we do things. So looking specifically at the Beluga, there are 3 sites. You can see these samples go back a long time, 40 years for this region. And that is, again, crucial to these international treaties. It's also crucial to providing information to you and to others on the Beluga levels of contaminants. In Pangerton and Senegilawak, we have 30 years of data there as well. And we also have a human health monitoring site in northern Nunavik, so we have samples from there as well. And Lisa's also going to talk a little bit about some of the data generated through other communities, through community-based monitoring programs or shorter research projects as well. So again, just thank you all for the hard work on all these years. I just really want to hammer home the point that NCP projects aren't just about science. It's about all these factors: communications, Indigenous knowledge, community engagement, and capacity sharing. If a project is strong scientifically but doesn't have these components, we will not fund it. And these components aren't assessed by scientists in the south. These are assessed by regions in the north. So So you get to decide what's a priority for you. And yeah, it's what makes us unique, but it also is what makes our work really relevant. Okay, this is my last slide. I'm just going to plug something we're going to talk about tomorrow. So we're starting up a assessment report of our own. It's called the Canadian Arctic Contaminants Assessment Report. It's a mouthful, so it's called the CACAR. This is similar to the AMAP assessments, but focuses on Canada. Our last assessment was 2017 and focused on human health. And usually our assessments in the past have focused specifically on contaminants work across the whole NCP. So it's a huge report, lots of information, good information, but you know, AMAP does a similar work. This next assessment is going to focus on one species, and we want it to be co-developed properly with experts and knowledge holders on Western science and Indigenous and traditional knowledge, so that there's not one chapter on Indigenous knowledge, it's throughout the entire assessment. And we want everybody to work together to really produce the best thing we can for the North. So maybe not surprisingly, the first species we've selected is beluga. And that's honestly largely because of the amazing work that's gone on here over the years, the collaborations you've all built with Lisa and other researchers, and amongst yourselves and everything else, and all the wonderful work that's gone on here. So thank you for being, honestly, what I consider a model project for the NCP. And I hope some of our other long-term projects will kind of follow this model and get stronger over the years as well. So as I said, there'll be an opportunity tomorrow to provide some feedback and input on what you'd like to see in that assessment. And I'm really looking forward to getting that input from you. That's the end of my part here. Please don't hesitate to come talk to me. We'll be around for questions. And you can go to our website as well for more information. So, thank you.
Thank you, Adam. I'm going to pick up from where Adam finished. And I realize we're running into coffee time. Is it okay to keep, do you want to break, Chair? Okay, good. So just picking up from where Adam left off, to be clear, the contaminants of concern are a lot of them are ones that accumulate in an organism over time. And so we're unable to eliminate it. Then what we also see is that those contaminants also increase in the food web. So animals that are feeding higher up in the food web will be at higher exposure and higher risk, so the polar bear. But also the beluga is higher on the food web level. So again, is an indicator species to us telling us what's going on. So what we do with the monitoring that happens out at Hendrickson Island is we're looking at levels of contaminants in them, and we're looking at how they change over time. We're asking a lot of questions about climate change. How is climate change interacting with the contaminants that we see in belugas? We know climate change is having an impact on food webs. It might also be having an impact on what beluga are choosing to eat or what is available to them. And so we're asking what that might mean. And lastly, we're asking questions about what the impacts are to belugas with contaminants. I'm going to put here my conclusion slide or some takeaways at the front, and then we'll kind of work through some details again at high level throughout the presentation. I do want to restate that the beluga Beaufort Sea population, the ones harvested here in the ISR, as well as by our Inupiat partners, remains one of the largest populations and one of the healthiest populations. For contaminant trends, what we have seen are trends in contaminants that we've been monitoring are declining or are remaining the same. We are not seeing increases in the contaminants we're monitoring. So we believe this has in part to do with those regulations, the banning of chemicals that are being put out there that we heard earlier from Adam, but also we're seeing influences from climate change and diet impacting how contaminants are going up and down. From a level perception, levels remain low and similar, so levels remain similar to concentrations that we see in other Arctic beluga populations and are low relative to belugas that live in urban areas. So I'll speak to that in a moment. With regards to impacts, we're not seeing any acute contaminant impacts, and in fact, what we're doing is using what we see here to inform and guide policy, and I'll speak to that. So this repeats some of what Adam said. NCP supports long-term beluga monitoring at 3 core sites. The project that I'm talking about is in collaboration with myself, other DFO scientists, Courtney Watt, who was unable to be here, Environment Canada scientists, we'll hear from next, Amila, and we come together, we bring all of our knowledge and expertise, and in partnership with 3 communities, Tuktoyaktuk, Pangerton, and Sanikiluaq. But by far there is a lot of leadership and partnership that happens here. I did want to draw attention to the fact that Paulatuk was also a real leader in identifying the need to monitor contaminants there. And NCP has supported contaminant monitoring in Paulatuk. And Paulatuk has been able to receive funds, and we've been supporting them to continue monitoring mercury. There is a poster about this project, as well as work in Paulatuk and other contaminants all along the walls that we can speak to in more detail. So there are other contaminants that have not been monitored over the long term, but this program has been a place to go and ask questions and almost be a surveillance for what other contaminants might be coming up of concern that we can take a look at. But this program has also supported other projects like the Country Food for Good Health that we'll hear from shortly from Sonia, and also other projects related to climate change and beluga health. We heard a little bit about mercury. Adam spoke to this. Mercury, unlike other contaminants that are man-made, mercury is a naturally occurring metal, similar to silver or gold. But our activities as humans and industrialization has increased the amount of mercury in the atmosphere by 2 and 3 times. And as Adam said, once it's entered the atmosphere, it can make its way to oceans and into our food web. Many of you who have been aware or are part of contaminant work are aware of mercury being a concern in the region. There had been some earlier work showing higher concentrations in the Beaufort. And this is something we wanted to and we continue to revisit. So some of the work done by my PhD student, Inuak Sedlovnik, who wasn't able to be here, wanted to dig into this a little bit more. So we partnered with the community of Arviat, who hunt a different population of beluga whales, but equally large, 60,000 belugas. I did want to acknowledge also Verna Pokiak here, who was a partner in that project. And what Inu wanted to do was look at how are beluga being sampled and how might that change the data and interpretation of contaminants. So this is really brief. There's also a poster here as well. But what she was able to do, and through learning, through culture and understanding that, as you know, you have a preference for the whales that you're landing, and the same is true for Arviat. And in Arviat, many of the whales that were being landed were smaller, younger, and also female. And so understanding that mix really changed how to interpret the data. She returned back to, that's right, and some of the whales landed, or some of the oldest whales, whales landed in Hendrickson or Barrie are much older, and so as you are older, you will have higher levels. But once Inu corrected for these differences of harvest preference, those differences that might have been seen before were really no longer prevalent. So levels that we're seeing are similar across the Arctic. But what about trends? So what I'm going to show you here are trends from the year 2000 to 2022 in mercury. That have taken into consideration the size of the whale and the age, because we need to consider that as we look over time. So I'm showing a subset of information for skin, muscle, and liver across medium-sized or young whales. And we see a general decline or a stable pattern. But I think what you might also see is that there's a lot of up and down in the mercury. It's not a straight decline. And so that has been a question of interest for us, is what is causing that up and down and what's driving those patterns? Some earlier work showed a relationship with climatic patterns happening in the Pacific region, and that those relationships or those oceanography trends were relating to what we were seeing. And so we need to, and we continue to dig deeper. But obviously, those climate trends have an impact on food webs. So of course, we have and we continue to look at beluga diet. As mentioned previously, their stomachs are empty, so they really challenge us in trying to understand what they eat. We use various ways of looking at their diet through measuring signals through their tissues. This is just one example here of a marker associated with eating Arctic cod in beluga, and all I want to show you here is it changes over time. So those changes are likely feeding into the trends that we see. But moving forward, something that we really want to drill down on is linking The work that we heard about yesterday in the Frosty, they're out there collecting those fish in key areas. So looking at the contaminants there and matching it very closely also with the tagging work, the telemetry work that we'll hear about this afternoon. So that is work ongoing and projects where we're looking more closely at those connections to understand those ups and downs. So to speak a little bit about other contaminants and long-term monitoring, the Stockholm Convention was a treaty to ban several contaminants. One of them that you might have heard of before is called PCBs, used in a lot of industry. And so I'm showing you a plot here showing a general decline, slight decline, but the good news is it's not an increase over time with this contaminant. But now I just want to switch gears and talk a little bit about contaminants that are new and emerging of concern, microplastics that we also heard about earlier. So in 2017 and 2018, we worked out at Hendrickson Island with a group of scientists to look at could we see microplastics in the stomach and in the intestines and in the feces of belugas. And so microplastics were detected. They looked at 7 whales. Microplastics were detected in all of the 7 whales at very, very low levels, but they were there. We also went on to look at fish to see if there were microplastics in fish, and there were at very, very low levels, quite low. But we did see, depending on the fish and where it lived, different types of microplastics. Again, there's posters also put on that we can take a look at. Another way to look at microplastics was to look at the chemicals associated with those microplastics. So a team of scientists took samples from livers and looked at, could we see microplastic associated chemicals in the liver of those same whales where we saw microplastics? The levels that they saw were either not detected or extremely low. So that is good news, that we're not seeing those chemicals, but work on this is ongoing. And it was good to be here and chatting with our collaborator in Alaska, Rafaela. And so they're also looking at microplastics in Arctic whales, bowheads, and seals, and they are detecting microplastics in multiple tissues. And so this is something that we want to take a look at too, and of course important for our discussion going forward, and also looking at how we use this information and knowledge to move policy forward. So some last few slides, drawing a comparison to the beluga that live in the St. Lawrence, so the St. Lawrence beluga. They're an endangered whale population. They live in an area full of shipping. They live in an area where there's industry dumping effluent into the areas. These whales do wash up on shore, not well. And back in the 70s, there was a high incidence of cancer in these whales, and it was associated with chemicals and contaminants being dumped directly in the river. One good news is we don't see the cancer there because there's been changes to what is allowed to be dumped into the river. Nonetheless, this population of whales are not doing very well, but the Arctic whales have served to support understanding healthy whales. There is a poster on this here, but what the Arctic whales have been able to do is provide guidance on setting thresholds and limits for what contaminant levels should be to reduce impacts that are seen in health in these whales. Just to note, the contaminant levels seen in the St. Lawrence range from 4 to 5 to 30 times higher and are of significant impact to those whales. So in summary, and for some takeaways, the trends in contaminants that we've been monitoring are going down, and we believe that's in part due to regulations and change. We are seeing interactions with climate change and feeding, of what beluga are eating and what levels we're seeing in the whales, and that's something we want to dive in deeper, but we're seeing those relationships, so climate change and contaminants are something to look at together. The levels seen here are similar to other Arctic areas, but significantly lower than what we see in contaminated areas like the St. Lawrence. We're not seeing any major acute impacts of these contaminants impacting the whales, and in fact, they're guiding policy on that for the St. Lawrence. I'll hand it over to our last contaminant speaker, okay. Anila.
Good morning, everyone. Is this okay? Yeah? Okay. So hi, I'm Amila De Silva, and Lisa asked me to speak about the forever chemicals, PFAS. Oh, you're holding up the 5-minute sign for me? Okay. So I wanted to start off just with this slide. PFAS are the forever chemicals called PFAS. They are regulated in Canada under various like government policies, but also internationally through the Stockholm Convention on Persistent Organic Pollutants. And in 2022, the Government of Canada proposed the listing of a particular class of PFAS for regulation, and there were these, this happened in Rome, and there were these nice like news reports that came out about the role of Inuit Circumpolar Council of Canada. I don't know if Herb's here today, but the Inuit were so instrumental in negotiating the terms of this regulation, and then further lobbied the different countries to ratify and sign off on regulating the chemicals. And why that's important is Adam talked about long-range transport of substances. So it's not enough for our government to regulate the chemicals. We need all the other countries to do the same when chemicals fly through the air and transport through the water. Lucy Gray, an Inuk from Nunavik, was in Rome. And this is, I've shown this picture here. This is what it looks like in those rooms. You see all the different delegates from the countries, and you have a little, kind of a space like this, and you push a button if you want to speak. And Lucy Gray made many interventions where she spoke on behalf of Inuit, not just in Canada, but around the world, and said things like this: Inuit have a right and will always fight to eat their highly nutritious traditional foods, free of chemicals, transported from faraway lands. And just, I want to mention as a Canadian, it's very moving. It was very moving for me to see that. And in a place where there are so many countries that are represented who don't know about Indigenous peoples and their way of life and how important these regulations are. So what are PFAS? I say PFAS, some people say PFAS. It's just an acronym for per and polyfluoroalkyl substances. They undergo long-range transport, but they're very persistent. They don't break down. Our bodies don't have natural mechanisms for breaking them down. And what are they used in? They're used in so many different things. I see a lot of the coffee cups here. Those are paper cups. What prevents the coffee from dripping out of that cup? Well, they've been treated with PFAS. A lot of weatherproof garments, outdoor clothing, have been treated with PFAS to prevent water seepage. They're also used in like silly things like cosmetics, like your waterproof mascara, nonstick cookware, paints, etc. And the big one is firefighting foams that are used to fight high temperature fuel-based fires, military bases. And PFAS are often in the news. Recently, this is our Minister of Environment and Climate Change Canada, Stephen Guilbeault, announcing further like restrictions on PFAS. So I've been part of Lisa's project since 2018, and we've been analyzing the liver of East Hendrickson Island beluga whales. This is total PFAS concentrations over time, and you can see just a decline in every year that we analyze. I also just want to mention, like, there's a lot of discussion about respect for the animal and respect for the environment yesterday, and I just want to say that when we receive these samples, we also really want to honor the efforts that were put into collecting those animals and the animal itself. It's very humbling to work with a specimen from a whale that has lived on this earth longer than me, and the contaminants that we're measuring have been exposed over their lifetimes. And those are things that are very important to our lab and what we do. Anyway, this is a very nice to see this decline. In 2019, you see that peak there. We're not really sure what happened that year. Lisa was just telling me that was one of the warmest years in this part of the Arctic, and that might have something to do with it. The other two populations there, Sanikiluaq and Pangatung, we are also seeing declines. And the most recent year there, 2023, this cursor might not work. You can see all the different locations are almost converging to the same concentration. So in terms of like the spatial differences, they're similar. So I mentioned we analyzed the liver. I realized that liver is not like an organ that is consumed from beluga, but this is the target organ where PFAS accumulate. PFAS bind to proteins, and that tends to be the liver. But Lisa received feedback from the community about, well, we want to know what else, where the PFAS are in the other organs. So we did analyze a few years for blubber, skin, and muscle. I know it's hard to see, but the blubber, skin, and muscle are under 10. They're like between 1 and 10. The liver shows like this high concentration of PFAS. Oh, sorry. And this is not my work, but St. Lawrence beluga liver have also been measured for PFAS in their livers, and it's much higher. So this is my last slide. I just want to emphasize we're going to continue monitoring PFAS in beluga whales. Yesterday, a lot of the hunters spoke about how climate change has impacted how they harvest, when they choose to go out, and like waiting for that cold weather. Climate change also affects contaminants and how they are released into the environment and transported. So in addition to looking at PFAS emissions in the world, we also need to understand how the climate is releasing and transporting PFAS that are exposed to the beluga, or that the beluga are exposed to. And that's it. Thank you.
Thank you very much, Amelia. It's always tough to go last in a group presentation because the others inevitably take up a little bit more time, but ultimately we're about 2 minutes into our coffee break here. I just want to give an opportunity if anyone has a really important question that they want to ask. Right here, okay. Before we get into coffee breaks, I think this is a really important subject.
Hi, my name is Gerald Iglangasak. I'm wondering, with the climate change opening up the Northwest Passage, what effects do the ships that are coming in? There's a lot more coming in now. Do they research or monitor with gray water dumping and dumping from the ships affecting the beluga whales? Because there's going to be a lot more ships. Just listening about St. Lawrence, and if we don't start right away, we're going to end up like St. Lawrence in about 30 years. Just asking a question. Thank you.
Do these work? Oh, they do. Okay, great. Thank you, Jerry. Thank you for the question. I think Jerry knows, or some folks know, I'm now in a slightly new position to focus on the St. Lawrence beluga whales, and some of the questions we just started to ask in this region was to look at PAHs, which is maybe an older chemical of interest, but one associated with vessels. So that's one thing we've just started to take a look at. Something we've not looked at, we've started to look at in the south are sort of like the gray water. There's something called scrubbers on vessels that are meant to clean exhaust, but end up releasing other chemicals into the system, mostly metals, but also PAHs. So PAHs are, I'm using acronyms, but are associated with oil and gas. So we're looking at developing monitoring programs to look at scrubbers and shipping in some of these contaminated areas. And so we've started one this year up in this region, but I'm happy to start looking at ways to look at those shipping associated chemicals, and we can put that on our radar and look at paths and ideas forward. Thanks, Jerry.
Thanks, Lisa, and thanks, Jerry, for that question.
So, Jason, I can just add to that quickly too. We again, I haven't looked at like gray water, but we do currently, or just finished funding a project that was looking at microplastics coming from vessel-based transit and things like that, which was a pretty unique study. They're still, I think, processing the data on that, but that is one thing we've looked at from a ship-based perspective.
Eileen Jacobson, I'm just curious, about the St. Lawrence beluga whales. Are they harvested down there? I mean, how did you get the samples like for the liver? I mean, you see how high it is compared to the north? Thank you.
Thank you, Eileen, for the question. And I do want to draw attention, both Emily, who you heard from earlier, is a veterinarian who works on them, and also Antoine Simon works on them. So they used to be commercially harvested, but there's no harvest of them right now, or there hasn't been, I think, since the 70s. But the samples that are being looked at are from whales that wash up dead on shore. And Emily, maybe you want to speak to the whales that wash up on shore. There is another program where they go and they collect biopsies. They'll follow the ones that are alive and take a biopsy sample to look at that. But as you know, there's no liver in the biopsy. But it's very unfortunate. The whales are not doing well.
Okay, thanks, Lisa. I think what we'll do is we'll take one more question from Stan, and then we'll have a break. And again, feel free to approach the presenters with your questions during the breaks and then the poster session afterwards. So, Stan, last question to you.
Lisa, you talk about management decisions when you look at all the studies you've been going. I never really saw anything about thresholds. Where do you set it, and when you do reach a point where you can make management decisions, how do you go about that? I mean, is it because the threshold has been exceeded or something?
That's a really good question, Stan. So for contaminants, there's been thresholds that have been kind of identified through, I'll say, very sick populations. And so they might identify, here's a group of dolphins who are unable to reproduce, and we see they also have a concentration of contaminants at this level. So a lot of the thresholds are set by some of these unwell populations. Through a partnership and Antoine's work, who has a poster, he developed a new threshold through using both populations and looking at the molecular level and looking at where genes were responding to contaminants. So we can take a look at that poster and we can share that. But something new that we're trying to do, it started just the last summer with a scientist named JP who has a poster, is to look at what's called in vitro. So we're collecting live cells with the help of the vets, collecting tissues and trying to keep those tissues alive. And then we can inject a contaminant and see how is that tissue responding, and then that sets thresholds. So that it's not great. It's because it's only one tissue. We'll see in other fish studies where you have an opportunity to put fish in a tank, they'll often use those studies to set thresholds where they can control the whole environment. But I guess it's a really complicated topic, and we can keep talking about it, but it's a very important one for management and policy. Yeah.
All right. You're going to... Okay.
Hello, can you hear me? There we go. I just wanted to add, so for Northern Contaminants Program projects as well, usually we see maybe if there's something like a higher concentration of mercury in a fish, for example, we don't see it in marine mammals quite as much, but if there's a level of concern that's kind of getting exceeded, then the researchers would flag that to us, and we would engage with the health authorities, who would then connect with the regional committees and do an assessment of that threshold and how many fish kind of exceeded, things like that. And there's a whole process that, again, the health department would take to assess that. And if it's something that they think is a bigger concern, then Health Canada would get involved as well. And then once they do their own risk assessment, then they reconnect with the health authorities, and they do the final kind of messaging out to the communities themselves. And again, the HTCs or HTOs would often do help with the messaging out to the communities to make sure everybody gets that information, but we leave it up to the regional health folks to do that messaging in the end.
Okay, thanks a lot, Adam, and that will actually lead into our next talk after the break quite nicely. So, round of applause for all the presenters. And thank you very much for all the questions. So we're at 25 after 10. Let's shorten the break a little bit to 10 minutes and come back at 10:35. All right, everyone. If I could get your attention, please. whether you're standing at the back or sitting at your seat. It's my pleasure now to introduce our next presentation on country foods for good health. For this session, we're going to be hearing from Sonia Ostertag from the University of Waterloo and Chloe Dolly from the Department of Health and Social Services. So, Sonia, take it away.
Good morning, everyone. It's just so nice to be here. And I titled the talk Building on the Outcomes of the 2016 Beluga Summit, because this project really is part of a conversation that began at the summit, and upon further reflection is really a conversation that started in 2006 when I started my PhD research with Frank and Nellie Pokiak and Lisa Lassetto, and I worked at Hendrickson Island, and I got to know the harvesters and started understanding how this work that we're doing on contaminants can become more meaningful and actually answer the questions that community members have. And that was raised at the Beluga Summit. And actually, what was also raised at the summit was how important it is to work with youth and to really engage with youth and support knowledge sharing between youth and elders. And so I think this work, as I go through it, you'll see how we've really tried to honor what we heard back in 2016 and really build on the success of that summit and all the work that's been done over the last really 40 years with the Beluga Monitoring Program. I wanted to really start by acknowledging that our research is a form of reconciliation. The data and samples that we've worked with are not owned by myself, they're owned by the Inuvialuit, by those of you in the room who are from the communities. And at the end of the project, all of the data will go back to the IRC and Joint Secretariat and will be able to be available for your use in future work. We're always updating our methods and processes. We work really closely with community research leads who we've actually hired and trained, and we've hired on a part-time basis over the entire project. And that way they can get that training, but also they become co-researchers on the work. And the priorities are really driven from the community first, and from I guess I would say from community members and the community leadership, and then region and territory. And we try to keep this conversation going so the work stays relevant. And this would be a good time to mention that unfortunately, Richard Grubin couldn't come. He had really hoped to be here, but he had to stay in Edmonton with his son who's going through some health issues there. And so he sends his regards, but I would say he's here in spirit. I've worked really closely with Richard since 2021. And this work on country foods, such an important topic for the region, wouldn't be possible without this huge team. So I'm only one person. I feel like I'm at the center where I get all the information and perspectives from so many different people, and I feel really privileged that I've been trusted with this work and guided by the community organizations and leadership and by community members. And then we have a lot of scientists and researchers in the South across Canada who help with the analysis of data and samples. And I wanted to acknowledge that Chloe Dolly is here with me today, and we've just started to work together, but our work has been partnered. So Country Foods for Good Health partners with the local region and territory. And so Chloe is here to support the work around messaging around country foods, because that is one of the responsibilities of the Department of Health and Social Services.
Just gonna gauge my distance from the mic. Can you hear me fine? So hi, I'm Chloe Dolly. I am a senior advisor with the Department of Health and Social Services. I work closely with Dr. Cami Candola at the Office of the Chief Public Health Officer and the Environmental Health Team. So I'm here representing both of those teams. I'll just go a little bit into the role and the responsibility of the Department of Health when it comes to this type of work. So the department is responsible for assessing the public health risk of a number of issues, not just contaminants work, but also giving advice on the impacts associated with environmental contaminants. And that's across the NWT. So some examples of how our department and the OCPHO gives advice includes health advisories, so that could be your boil water advisory or your do not consume water advisory, but also the general health advisories like the site-specific fish consumption advisories. And basically there are tools to relay information to the people that need to know the information in order to manage their risk so that they can take steps to protect their own health. So the department doesn't conduct primary research like the research that you see posters on the walls or that we're talking about this week, but we do engage and collaborate with researchers like Sonia's team to make sure that the message being communicated with the results makes sense and is protective of health.
So the question we're really trying to answer in our project is, what does a healthy diet look like at all stages of life? And most importantly, the Inuvialuit harvesters and knowledge holders and youth have very important perspectives on this. And it's really important that that is balanced with the Western science that has a different perspective on food and health. And then public health is involved in that messaging and interpretation. And so by working together across community science, Western science, and public health, we hope to be able to answer this question. But it's good to start with a quote from an adult in a Klavik who said, "I think traditional foods are a big part of our local diet and are healthier for us in the community to use over store-bought food. It would be good if all community members have access to all traditional food for more healthier living." And this is really the perspective we hear when we talk with community members and elders, that the country foods are really healthy and really important for health. So we started the project by working with, we collected samples, but we also then added a piece around working with elders and youth. And what elders said was, if you want to live a good life, you'll have to continue eating from the land and keeping out on the land, because the land heals you, because the food out there is healthy. And so this is the story. And it's important to recognize that this is true even if we didn't record it and study that as researchers. But it is really helpful when we're talking about food that we can have these different perspectives in the same presentation, in the same space, so that we don't lose sight of that knowledge and perspective coming from your knowledge holders. We also heard another elder say, "I think the government should really protect our caribou no matter what. Since I was a little girl, caribou was around here. We're going to keep on doing that for our grandchildren, for my great-grandboys to continue learning from their uncles and keep on doing what we're doing. If we run out of caribou and things like that, it's going to be a hard time. We're going to suffer, and we don't have the money to buy all the junk food and all the things." So this idea of how important your country foods are for your health is once again reinforced. When we worked with youth, so I was working with Maria Ramirez Prieto. She's a PhD student, and she worked in all 6 communities with research leads and youth as co-researchers, and they used photos as a way of sharing their knowledge and perspectives, because sometimes youth can be really quiet, and using photos as a way to let them have space and share what they're thinking. And so the youth in the 6 communities took photos, and then they were interviewed to share why they took those photos. And then they also had a chance to sit with elders in each of their communities to talk about their thoughts on country foods, so that they could have a chance to sort of share their perspectives. And what they said was that the foods are not only nutritious, but they're really important for mental health and their pride and confidence that it's part of protecting the land and passing on knowledge. And this is probably something you already know as a Inuvialuit, but it was so nice to hear this said in such a way and to really reinforce how important these foods are for all aspects of health. So one of the youth from Inuvik said, "The elders teach you how to harvest right, and just like how my father taught me, I'm going to keep on teaching my children or future generations. Just even as a teenager, I know a lot about it, and I think more kids my age should be out on the land. It's pretty fun." And so once again, this really important piece around young people are already sharing their knowledge, and they're proud of that, and that's something to keep creating space for. And finally, Maria worked with families in 3 communities in Aklavik, Tuktiaktuk, and Ulahaktuk to look at how families can support being on the land and what the importance is within the family. And the themes that came up were around healing, going on the land as a way to access the country foods, but also to learn language and share the language and just have a nice time, enjoy an enjoyable time together. And that's something that I think we've talked about a little bit and something that I've heard, and it's not in the presentation, but I think it came up yesterday. Not only is it important to go out for a couple of days, but getting out for those extended periods of time for these benefits to be really experienced. So since we're at the Beluga Summit, I'm going to shift to like Beluga focused. The Beluga focus, so overall, if we're thinking about what foods people are eating in the communities, we have older data from 2007, 2008, showing more than half of the people who participated in the study, the Inuit Health Survey, were eating beluga oil and dry meat. And then more recently, we did a survey in the schools in the ISR. So in each community, I worked with a student, Alyssa Salins, and she was able to do a survey in each school to find out what do the students want to eat and what activities do they want to be involved in. And generally, more than half, or around half, wanted to eat beluga. So it's still really important food for students to be enjoying. And I did want to note that in Inuvik, we weren't just focused on Inuvialuit students, and so this lower percentage might be really because the population is not all Inuvialuit. There's a mixed population here in Inuvik, but overall, there's still this really strong interest in beluga. But it's also important to note, there's an interest in all of the country foods, essentially, in eating those foods and also being involved in harvesting, but I just focused on this for today's presentation. And the perspectives on traditional foods based on the survey that we did with community members across the ISR, everybody who responded felt that traditional foods are the best for themselves and their family, and they feel comfortable preparing traditional foods safely and choosing traditional foods that are nutritious for themselves and their family. And then a smaller percentage, of around less than 40%, had concerns about the quality or safety of some or all of the traditional foods that I eat. So I think overall, once again, we're talking about a lot of heavy things like today around the science of beluga whales, so those analyses that are happening. And it's important that the work we're doing around contaminants doesn't disrupt this sense of how healthy these foods are and the confidence that people have in these foods, and to just let you know that the work we've done has shown that there's no reason to be concerned. We're not concerned or alarmed by the findings, but it's important how we communicate that information, that it doesn't create fear. And although people are confident in country foods, the survey responses showed that there were some concerns around quality or safety of fish, beluga, caribou, and birds. And so there are some concerns that can be responded to. And there were concerns around tapeworm and botulism, which we heard a lot about yesterday. And overall, there's a really strong interest in learning more about what is in both country foods and store-bought foods, the effects of food preparation, how do you access and prepare the foods, how much is a healthy amount to eat, and then specifically for store-bought foods, what store-bought foods are healthy to eat during pregnancy and for children to be eating. So to help with answering those specific questions, we were able to sample more than 700 foods from about 210 food types. So those are raw and prepared. And we had hoped to receive samples of prepared and raw samples of foods in the community, but most of what we got was raw. And so to be able to answer the questions around what's in the food, we had to prepare them in the lab. So we did the best we could at like drying the fish and drying the meat and cooking it in different ways. And what we found, this is a really busy slide, is as you all know as harvesters, that your foods are very, very nutritious. And so this just shows you how all of the different animals and actually parts of different animals can provide so many different nutrients that are important for health. And then I took this information, and a few weeks ago we made these posters over here that are just like a starting point, but they really show you how the different foods, what vitamins and minerals are in them. And if you wanted, you could compare to the label on a food item at the store. So that's like a starting point. I would love to get feedback on how else we can share this information. We also found a really special group of antioxidants in muktuk. So seleninine and ergothioneine they help with inflammation, so for heart and brain health. And the muktuk actually has the most of this seleninine, which is a very long word, but basically what's special about seleninine is it's thought to actually help with protecting you from mercury. So when there's mercury in the food, seleninine can help keep you from having any effects from the mercury. And you can see that black bar is the biggest for the muktuk, and actually the ergothioneine, which is just a general antioxidant, it's also very high. So the muktuk is a bit of a superfood, I would say. And it's actually highest in the whale tail based on work in Nunavik. So that's where this project sort of was conceptualized. And I thought it was really neat yesterday that there's a song about the whale tail that the Nunavik drummers and dancers were sharing with us. And so this idea of the whale tail as being this important food, and then it has this really high level of seleninine is really interesting and protective. So when we're thinking about the risks of contaminants, one of the reasons we want to think about risks is because of those questions coming from communities based on all the monitoring that happens. So there's a lot of information on posters and in presentations about contaminants. And so we wanted to make sure we were able to share whether there's a reason to be concerned. And so we looked at heavy metals, forever chemicals, and then legacy chemicals. And we did a risk assessment for these different chemicals. And it's a balancing act. We look at how much do we think people are eating of the different foods, then we multiply that by what is in the food, and then we compare that to an amount that's considered to be safe or low risk for health. I do want to acknowledge that the data we use on how much people are eating of the different foods is very old. The most recent data we have is from 2007, 2008. So people's habits might have changed since then, and so we are working on a survey to update those numbers. But what we found was the risk of lead exposure is low, or the health risks from lead in food is low, but we did find higher amounts of lead in some of the foods, especially those small animals that might be hunted with lead ammunition. So eider duck liver, hare kidney liver, ptarmigan liver and fat, and the snow goose and swan liver. And so we know that lead exposure can be higher in country foods that are hunted with lead shot. And especially if the animal's not cleaned right after carefully. But even if it's cleaned, that lead can go into the rest of the animal. And we know that lead can affect brain development and function. So it's something to avoid. And so the recommendation, or it's good to think about that the steel and bismuth or iron shot are going to lead to no lead in your food. And I think it's an important conversation because I don't understand like what decisions or like how those decisions are made around lead ammunition. And so I think would be maybe something really useful to hear about today, later on, why lead shot might be used and if the alternatives work or don't, or maybe they're more expensive, and how those decisions can be supported to use the non-lead ammunition. We looked at cadmium in country foods, and it can cause kidney problems and lung problems, and it's also carcinogenic, so it's been linked to cancer. And what we found was the amounts in the food are, expected to be of low risk for health effects. And actually, previous work showed really that the smoking is what's causing cadmium exposure in the ISR. And so the cadmium in cigarettes is a much bigger concern than the cadmium in country foods. Finally, the mercury risk assessment. So mercury is of a risk for the developing fetus and young children. So the brain, as it develops, is very sensitive to mercury. But we found that the health risks are expected to be low for mercury for most Inuvialuit, including women of childbearing age. And when we compare that to the data from the work that was done in the Inuit Health Survey, the numbers really matched. So most women of childbearing age, so 85%, were below a level of risk. So they're at very low risk, and only that 15% was at that higher level of risk. And so it will be good to update that information of what people are eating today, but also to look at what kind of guidance to give to women in that group who might be at the risk of having a little bit more mercury from their diet. Our PFAS that Amelia was talking about, we know they're decreasing in Beluga, and we did find us in under 10% of the foods was a higher amount of PFAS, but when we look at how much people are eating, the risks are expected to be low, so that's also good news. And then for PCBs, which is one of the groups of legacy contaminants, we found those levels are, from Lisa's work, they're stable or decreasing. And then when we look at how much of those foods people are eating, once again, the risks are expected to be low. So based on this information around contaminants, the risks from contaminants, and then all the benefits from the nutrients and all the value and knowledge shared from Inuvialuit knowledge holders, we need to work together to co-develop messages to support healthy diets, not just focused on contaminants, but all those health benefits from eating country foods and being out on the land. And that's something that I'm really hoping to do over the next year, but probably more, to bring everyone together to help make those messages really relevant so that people in the communities can make those healthy decisions and feel confident that they know that they don't need to worry about contaminants, that they have the information they need, and that they know that the food is nutritious that they're eating.
So I've only been in my current position, for a short time, but I've been with the Environmental Health Unit for several years, and I was raised in the Northwest Territories. I can appreciate that historically maybe messaging from the GNWT Health and other departments has not been co-developed and has not had a relevant and impactful message. And I just want to highlight one of the messages from the Environmental Contaminants General Info Sheets, where it really is highlighting that traditional or country foods are healthy, they're beneficial, and there is a low risk to consuming a variety of these foods. In the case of higher risk for a specific animal or a specific part of an animal, or for a specific consumer, like pregnant women or children, advisories are the tool that our department uses. Adam touched on that a little bit. But this is the kind of message that has historically gone out, and my role is to work with researchers and the NCP and our team to develop messaging that is relevant, and it needs to be a collaborative approach. And I acknowledge that Inuvialuit knowledge and perspectives is the foundation of all of that. And so in my role, I will work towards better communication of messaging. And one of the pieces is working with researchers like Sonia and incorporating that into our messaging.
And one of the things that's really helpful in developing the messages and even how we share the messages is that we had this survey, the Inuvialuit Food Messages Survey, that I kind of referred to a little bit in the presentation. But we were asking community members specifically, who do you trust and how do you want to learn about food and diet? And so I put in bold, the elders, family, and friends are most trusted. And then other groups are also trusted. And so those are the groups that we want to work with in terms of sharing messages about food. And then we also want to use methods of sharing information that are preferred. So finding ways of getting the information to on-the-land programs, using small group discussions and food-based programs, rather than just pamphlets and phone calls and TV and radio. So in purple at the bottom, those are not preferred methods. Those are often used because maybe they're really convenient, but they're not preferred. And so maybe finding those alternate ways and finding ways to work with elders and knowledge holders and harvesters so that we have really clear messages about country food safety, and then that way it's the same message being shared by everybody. And so finding a way to get everybody on the same page, which I think we're actually really close, but finding a way to talk with each other more so that information can come back from the trusted sources. So overall, strengthening partnerships to co-develop communication materials is really important. I have a photo here of the cookbooks we made with the schools, and so we worked with schools on cookbooks to highlight the different recipes in the communities. But I think coming here with Chloe and having a chance to meet with Jen on Friday, and hopefully having chances to meet with your leadership here in Nuvik, that's a really important first step to that strengthening partnerships. Engaging with the local knowledge holders and health centers. This didn't really come up in the presentation too much, but students want to eat more country foods and they have a lot of interest in country foods. So being able to serve those country foods in schools would really improve the quality of the food in the schools, because we know how nutritious the foods are from this area. And also reinforces that these are healthy foods. And so I think it's really important also that in the hospitals and in different food programs, finding ways to serve the country foods so it's not always using a PO at Stanton because that's the sort of way it's been done, but finding ways to get those country foods in your programs. Continuing to support knowledge sharing and on-the-land programs and community harvest. Advocate for those policies so that we can reduce contaminants even further in the environment. And really, when we talk about country foods and healthy diets, I would be remiss if I wasn't acknowledging the food insecurity issues of Inuit Nanungat and the ISR. And so really looking at those drivers and finding a way to resolve those longstanding issues so that people have the choice in what they're eating and they're getting enough to eat, and that your communities are supported and people are not having to make decisions to cut back on what they're eating or not able to decide on their preferred food because they don't have that opportunity. So our next steps are co-developing the messages, completing the dietary survey, and in Ulahaktak we have a few more samples that we're processing and hopefully also going to collect, and then communicating our results. So we have these posters that we're going to hopefully turn into a booklet, maybe a website, and we are trying to make food calendars with any of the communities that are interested. So we have funding to make a food calendar that can complement the Canada Food Guide, which is a very Western approach to looking at food, and when I look at it, I don't know how that can be relevant here in the ISR. So having something that really illustrates the wonderful foods and culture here. And I guess the key messages are that our work really reinforces, so the analysis of country foods and the reinforces your traditional knowledge and values. I think it's important to highlight how knowledgeable your youth are, how trusted elders are in your communities, and that the advice that we work on together, that it has to acknowledge those local realities and it can't just come from Waterloo or Yellowknife. Thank you very much.
Thank you, Sonia and Chloe, for addressing that incredibly important topic and such a great presentation. So we actually have quite a bit of time for questions here, which was by design. And so I'll open the floor up to some questions.
Hi. Oh, wow, that's loud. I'm Ryan Lenny from Nuvik. I see a lot of the contamination graphs, and they're little, and then slightly bigger. I feel like to better convey the message, you need chicken nuggets at the end, or farm salmon, and just showing the farm salmon way up and we're way down here. Because like a lot of your graphs they're all small, they're all negligible. But just having one bar slightly bigger, people are like, oh, I don't want to eat that because that's worse than this. But really, they're all not bad compared to store-bought food. That's it.
That's a really good point. Thank you. And on the posters, we did try to show, like for the nutrients, like that comparison of like the high quality of the traditional foods and then what you would get at the store instead. But I'd be happy to, like the contaminants are a little bit maybe more challenging in a way because within the wildlife population, animals that are eating higher in the food web are going to have higher concentrations than a chicken or a piece of beef. But then there are also all those benefits, and as long as the risks are low, I agree with what you're saying, but I think sometimes if we look too closely at the graph, we lose sight of also the bigger picture that the risks are considered to be low, and so the actual number, and I think that's why we wanted to have the work with elders and youth and the nutrients, because then the whole story becomes that these foods are so incredibly nutritious and important for like overall health. And as long as the contaminant levels are below concern, then that is the message. But I hear what you're saying, and I'd be happy to make some of those graphs as well to convey that. But I wouldn't want to show the country food having higher concentration of a contaminant than a chicken nugget, and then it doesn't mean that the chicken nugget is better, but there's probably a way to do that, and I appreciate the comment. Thank you.
Okay, we had a question over here.
Daryl Nasagelowak. When we talk about lead in geese, I think if we haven't, we should ask whether the goose was harvested with steel shot or lead shot. The amount of steel shot sold today is really high. For 150 years, probably people all over the south in the winter range have been using lead, and there may be a lot of lead in the areas where our geese winter. So if we weed out or actually ask the question that we want some samples from geese that were shot with lead and some that were shot with steel. And you asked the question about, why lead? Lead is maintains its velocity better, so it starts off slower than most steel shot, but it keeps its energy further, so it's more efficient as a shotgun shell. You rarely find, in my experience anyway, geese that survive lead shot, but the majority of geese that I've harvested that were wounded and healed over again had steel shot in them, and you could tell it's steel because there's a big black ball inside the goose. At the core of it is a steel shot. So I think that moving forward, if we continue the research that we ask for steel shot geese and lead shot geese, and see if there's a difference, whether they picked it up in the environment or from being shot with lead. Thank you.
Yeah, that's a really good point. And it was interesting to me that none of the muscle had these higher concentrations of lead, so it was really seemed to be concentrated in the organs. And so that's something that just raises a question as well, like, is it coming from the lead shot or from the environment? But I was, I guess, curious, and when I went to I was in Tuck and Sachs Harbor a few weeks ago, and I looked, and I mean, the lead shot is for sale in the store. So I think there's a real value in looking at what is in the birds, depending on how they're shot. But I also think there's value in looking at, like, why is it that the lead shot is working better than the steel shot, and how can the steel shot work at the level needed so that it and be the same price, and so then it becomes a non-issue, because if the steel shot is safer, but if it's not working, then it's not a good option. So I appreciate that, and I think, like, it's a real conversation, because once again, putting a message out don't use lead shot, and then that's not really feasible, it's not really useful. And I agree, like knowing more about how much of the lead is in the muscle of the birds if they're hunted with lead shot. But I think the organs are also eaten, so I guess it's something where it would have been good to know those ones, and maybe we can have a few more samples from the spring from Tuck. We have funding for some more analysis, so maybe we can get some geese from the hunters and to do that last step.
Thanks, Sonia. James?
Yes, thank you. Good presentation, Sonia. But anyway, I'm a firm believer in the foods that we harvest and consume. We've been doing that for centuries, and one only has to look back to the late 1800s, early 1900s, when a lot of these explorers, fur traders, trappers, whatnot, many of them wouldn't have survived without the local people in the area. So to me, you go back that far, and we've been consuming these country foods for centuries. So I really believe that's the most healthiest food you can have. But I'm very happy with all these research is being done on the species that we consume. Thank you.
Thanks, James.
Oh, I just wanted to elaborate a little more on what Daryl said. Geese and geese shots, maybe in the fall and in the spring, 2 different seasons. Not too many people harvest geese in the fall anymore, but maybe there might be a difference in contaminant levels from the spring and in the fall. Thank you.
And I think, I'm just wondering, it might just be worth looking at lead in particular, because that's the one that came out for geese. Everything else looks great. So I think that idea of, maybe having a chance to get those samples from Tuck. And I don't know, in Paulatuck, are there any fall geese harvested in any of the other communities? Because anyway, we can talk about whether we can get some from spring, fall, and we could quickly get those analyzed for lead if that's really straightforward.
Thanks, Sonia. I was wondering, we do a lot of research with our beluga whale in the Cogmalic Bay area, and as far as we know through research and that, the beluga whales winter in the Chukchi Sea, and I'm just wondering if anybody does any research on that and where the wintering range is, and see the difference in all the samples you're taking there for whether it be mercury or plastics or whatever, or it could be pretty high over there, and maybe their contaminants are kind of dying off by the time they come this way, or vice versa.
That's a really good point. I know, so first of all, with mercury, we know that those concentrations in muscle are probably changing quite quickly over the year. So in the span of months, those levels can change. So that's a really good point. I don't actually know, Lisa can answer that, or if Raphael is here, but Lisa will help with answering that question. Thanks.
Good point that they are wintering in the Chukchi. We collaborators started collecting fish samples from the Chukchi to compare the prey. So we are seeing for mercury lower concentrations in the Bering Sea, but we're working with North Slope Borough to share samples, and we'll be analyzing them to look at contaminants that will give us some clues.
And the difference between the Beaufort and the Chukchi and the Bering Strait, we actually have it in polar bears as well. So the southern Beaufort polar bears are different than the Chukchi polar bears. And in general, we think there are higher ones over on the Beaufort side. And we're not quite sure if it's just diet change or it is the way there's runoffs or whatever it is. But there is a signature difference between them, actually. Thanks. But yes, we definitely are looking at it. And since we're sharing, the animals are sharing the ocean and people are sharing the food. So yes, we are trying to collaborate on those things and fill the gaps.
Hello? Okay, sorry. Just wanted to comment my veterinary perspective on lead, as we do look at lead intoxication in wildlife. So basically when we see intoxication in wildlife, so it means that the lead has been incorporated in the system. The animals need to actually eat the lead, which is usually from lead pellets making their way into the environment after hunting, or eating a carcass that's been of an animal that's been killed with lead pellets. If the animal has been shot and survived and has lead pellets within the muscle, it will stay in there and would only then affect a species that would eat the muscle with the lead, if that can help.
Oh, just putting up my hand for, I could give you some fall geese, if you're asking. I like hunting in fall.
That's very generous.
Thank you.
Looking for other hands.
Hello. Just touching on your fall geese hunt. Ulukhoktok does do a lot of fall geese hunting. I just wanted to say that.
Juana, thank you. And that's great because we'll set up that sampling then and talk with the HTC in June about that.
Yeah, I just wanted to make a comment about human health contaminants. Is anybody doing any research, and are you collaborating with people on the human contaminants?
The levels in humans. And so that's been a gap, I'll say. So when I started my work on country foods back in 2018, we were starting this project, and the Hanuipita National Inuit Health Survey was getting set up, and they weren't sure if they would do the biomonitoring, so the analysis of human blood samples and tissue. They're not doing that, and so it's definitely, I would say, a gap, and we haven't heard a lot of interest in doing that type of work, but we're open to that. I collaborate with Brian Laird at University of Waterloo, and he's very experienced in human biomonitoring, and they did that a lot in other parts of Northwest Territories and Yukon. So if there's an interest, we could look into that, but we haven't heard a lot of interest in that, and it's a lot of work and a lot of time, and I think we would want to know what the questions are, but I'm definitely open to supporting that if that's an interest.
Just one more, Sonia. Like for seasons, like all our animals and mammals, they have different feeding seasons. And that I know, like you brought up the caribou and that, and summertime you see them eating wet vegetation and leaves and that off the willows and that, and in the wintertime they don't have any of that. So I was wondering if you had actually take samples from the winter harvest of the caribou and that, because then at that time they're probably only eating lichen or that kind of vegetation, and how would that be different? I mean, if you could see the difference from summer or fall to winter time and that, and then if something did bad come up here, then we'd know what time it would be. They'd be getting rid of that contaminant versus getting it.
Yes, that's a really good point. And I think like this initial part of the project was like doing a snapshot of the big picture, and then these questions around the geese and lead and the caribou and the seasonal diet of the caribou. I think those are really interesting follow-ups. I know that there's that NCP project through the HTC on the caribou, and I think that's like a fall-winter project with Mary Gamberg. But it would be interesting to see what she's finding, and then I can look back at what season the caribou were hunted that we have so far. But they're from all the different communities, so we don't have a lot of samples per community. So if that's of interest, I was gonna add actually, I'm staying at the Research Institute, like at the ARI housing, and the people I'm staying with are researching the sumps. And not to like raise alarms, but a lot of you who live in like on the mainland, and you're in like Tuckananuvik and Aklavik probably know about those sumps, those waste piles from industry, and how those are thawing. And to me, that is really interesting. And when I talked, well, interesting in a morbid way, like I'm not happy to see it. And I think there might be something there as well around, like the animals are using these warming areas of permafrost that are warming because of the sumps, the waste that's buried there. And it's creating microclimates, and then the animals are using those spaces. So I would say maybe that type of analysis might be really useful. I don't know how one would do it, but this idea that like there are a lot of ptarmigan, and then there are moose tracks, like a lot of animal tracks in that area. And so I was talking with them just out of curiosity a couple days ago, but I just wanted to raise that. So you might have heard about it already, that there's this work going on, but this idea of like those sumps and how those habitats then might be used by your wildlife. I think that was a question that was raised that kind of builds on what you're talking about, Jim, depending on the time of year.
Hi, Sonia. Thanks for that presentation as well. Just on a question of fall hunts for the geese, I just wanted to give you some insight, I guess. At one time, I think it was considered TK that we hunted when it tasted or were in the best condition. I remember a time when we used to hunt geese in the fall, but not to store them for the winter. It was because we used to be out camping and hunting in the fall time, fishing, and so for your daily needs, we'd, have whatever was out there, and geese in the fall time used to be heading back south. Today, it's out of necessity, I guess. If we didn't get a spring hunt that'll support your winter, you might supplement it with some fall hunt. But it's not the best because of the fresh feed they've been having, and it doesn't taste, as fresh as the spring hunt. I think back as well, a little while back, the question of legality in spring hunts used to be an issue, and now with their population, it's no longer an issue. But again, I as well think that in terms of science and testing and for health, that might be an idea to expand your studies to the fall hunt, because there are a lot of times that, I think we got an answer about how this lead contaminant is brought on from one to the other, but just to detect the amount of lead or pellets are in the geese at times and be able to gauge that. We've been hunting every year for millennia, and I can't imagine how much lead is out there in the tundra, hard to imagine. Thank you.
Thanks, Ray. Any other questions?
We have time for one more question.
Brian Wade has a question in the back.
Brian Wade.
Full name, Brian Wade.
Check. Hey, I just wanted to, more of a statement in regards to, how he's telling me to move over. More of a statement in regards to the approach on country food. Country food is not a plan B, it's not a secondary diet to Inuvialuit. So when you compare store-bought foods to country foods, we need to compare both and the risks on both. And I know that Sonia, through an engagement session last year, we looked at produce, right? And the contaminants and the preservatives that are in there. So when we look at country food and the approach that Inuvialuit take on it, it's a second option to us. If there was a table full of country food and store-bought items, I guarantee that the country food would be gone first. So taking that approach to look that it's not a luxury, it's a primary diet to us. And then when you look at contaminants in country food compared to preservatives and hormones and steroids in store-bought foods, what's the lesser of two evils, right? So that was just my comment. Thank you.
Yes, thanks, Brian. And I think I didn't have a chance to share a lot about store-bought foods. Like there's a little bit there, and that's really comparing sort of a substitute. If you weren't able to get the country food, what would be a substitute? But what we know is that when we go to the grocery store in the ISR, you're not always able to get steelhead trout from the south, and you're given an option of a lot of processed foods, and we know that those are essentially toxic. They are really, really unhealthy. So The sugar, the saturated fat, the salt, it's really unhealthy, and it's really hard to manage chronic conditions like diabetes when you have those foods available. And so once again, yeah, the country foods are the gold standard in terms of the healthy foods you have available, and the alternates that are comparable aren't as good for you, and then what is actually going into the cart is often worse because it's those processed foods. And I think like those questions around like why are those foods being purchased and used so much, that goes back to food insecurity, and I think like the bigger issues there. But I think the fact that you have this abundance of food on the land, in the ocean, in the lakes, and the rivers, it's such a privilege, and it comes from your stewardship of this area. And I just think it's such a privilege that it's there, and like Brian said, that's your first choice. And I think, you know, the more that can be reinforced and the more that those foods can be made available, the better. And actually what we've heard from the surveys that we're doing now, the diet surveys, is people want more country foods than they're getting. And so looking at how that can be done, I think will be really useful.
Okay, thanks, Sonia. We'll take one more question from Daryl here, and then I'd like to get into the posters and make sure that we have enough time for the presenters there.
Thank you. One more, thanks for your presentation. One more really quick comment. There was some talk about maybe they were picking up the pellets while they're feeding. When the geese come north, they're feeding on the berries that were frozen all winter. So their primary food source when they reach us is, as the snow recedes, they're going north. So they're not able to get at the roots yet, so they're not going to be picking up the lead shot that way. We do know that when we're cleaning the geese, there's some different shoots and stuff that they're eating further south. Maybe also we should collect whole geese so that we can check like the contents of what they've been eating that day, or that, you know, they come a long ways in a single day. When they do reach us, until they reach their I guess summer range, they're not going to be rooting, but we're not hunting in those areas. So when we're shooting these lead shot all over I guess you can consider the flyway, they're not going to be picking the lead shot that we're shooting, because they're primarily feeding on the berries as the snow recedes. If you're quoting ITH all the way to Tuck, there'll be thousands and thousands of geese on both sides of the highway where the dust flew and melted the snow, and they're feeding on the berries. They're not in those, they're not able to root yet, to pull the roots. So, yeah, that's my comment.
Thanks, Daryl. There's some music playing here. I think that means it's time to change sessions. Okay, so we're now going to move on to the so-called elevator pitches or lightning talks. This is an opportunity for the poster presenters to pique your interest and to tempt you to come and see the poster at the break or during the poster session. So could I ask all of the poster presenters to come and stand by your poster? And while the presenters are... Thank you, Daryl. I should have done that. Okay, one thing I just wanted to point out before we start these lightning talks is, you know, we've heard a lot about future leaders among the communities. Nuvialu Future Leaders. We also have a lot of students involved in this research, and they're really our science future leaders. And a lot of this work that you're seeing here is thanks to the hard work of a lot of students. Unfortunately, I don't think there's a lot of students here right now, but I just wanted to acknowledge the work of students that without which we wouldn't have all of this fantastic science. So I'm going to start over here. And so Shannon can go first and set the example.
Hi everyone, I'm Shannon McPhee. I have two posters here that I'm excited to share with you. So the first poster here is about the design of a new way to tag beluga whales that's remote, so we don't have to capture them. And it can be hunter deployed and is less invasive because there's no capture and there's one single attachment point. And this method is based on inuvialuit harpoon techniques. And it's something that was developed together with scientists and community members working together in the field and really applying inuvialuit knowledge, engineering, design, and science to try to come up with a new way of gathering information about the movement of beluga whales that's more flexible and less invasive. So my co-presenter is Dennis Airy, and there's other people here who are involved in the work. Please come talk to me. I have a second poster about unusual marine mammals. So we've heard a lot of observations from harvesters about different species that they're seeing that aren't as common in the area. Like killer whales is a good example. And I've got a poster over there of the records that we have from harvester observations and scientific records of when and where these different marine mammals have occurred in the ISR. And I'm excited to hear your feedback on other observations that you might be willing to share and how to capture this information.
Thank you, Shannon. Natasha.
Thank you, Jason. Hi, everyone. I'm Natasha Neves. I work with Environment and Climate Change Canada. I'm a research technician, and I have two posters today about microplastic pollution. And most of this work was done by Madeleine Bourdage, which some of you may have worked with. She just had a baby, so she couldn't be here. And I'm very honored to be presenting this in her place. My first poster is about the 2022 beluga harvest. We analyzed some stomachs and looked into microplastics in them. We did find some. They were very similar levels from the 2017 one that Lisa showed before. So overall, these numbers were very low. The second poster I have is about microplastics in the environment. And we have 3 different projects trying to understand where and how these microplastics may be getting there. So we have results from Great Slave Lake and also the Mackenzie River. We also have another work around Zed Creek. So trying to understand if the highway may be a local source of microplastics to the region. And the third project is the one about Arctic shipping risks. So this was done in Nunavut, in Arviat and Pond. So I'm also happy to talk more about that later. Overall, all of these levels were relatively low, which is good, but there were some differences that I'll be happy to talk more later. I'll also have a microscope with a sample if you want to look at what microplastics look like later. I'll be happy to show that. And yeah, thank you.
Hello, everybody. I'm Cody Malone. I'm a PhD student at the University of Saskatchewan. And we tested a bunch of belugas from 2014 to 2024 for 3 different parasites. And what we found is the 2 parasites of greatest concern, Trichinella and Toxoplasma, were at either 0% or extremely low percentage, which is excellent news. So if you guys want to learn more about the parasites we tested, please come see me. And I would be really interested to hear about your interest in parasites and disease and what you think would be good areas for future research. Thank you.
Hi everyone, you've heard from me already today. There's a couple of posters on contaminants, but I want to draw your attention to, there's a poster behind the speaker that I'll move maybe over there, and that's by a student looking at some of the whales from Paulatuck and Hendrickson, but whales that were identified as skinny and of concern, so that's one. And then there's another one I think back there looking at the stress hormone called cortisol, and so I draw your attention there. In particular, I wanted to connect with some folks from Aklavik and Sax Harbor to just talk through some of the results there. And then I'm here to address any contaminant questions, and unfortunately some of the students weren't able to join.
Hi again, everyone. I have a poster here, lots of colors on it. It's on the PFAS chemicals in the beluga liver. I showed some of the data today in my talk, but I'm also happy to talk about trends in males versus females, adults versus pups. Come by my poster. I'm happy to talk more.
Hello everyone, my name is Antoine. So I currently work for Simon Fraser University in Vancouver and the University of Montreal. And here the work I'm presenting, so it's all about the sonorous history of beluga whales, but it's most of all how the symbols you provide us can help understand why that population, endangered population, is doing not too well. And so here I present some very interesting results. That shows, so we use your sample as a reference study, as a reference populations, because they're not exposed to high levels of contaminants. And we show that the sonorous history beluga whales have an impact on their immune system, and they they struggle probably defending themselves against all the stress they face. So yeah.
I'd also like to point out Antoine's fantastic shirt. You can't see from back there, but it's covered in little belugas. Well done.
Hi, I'm Amelia Dalpe. I'm a veterinarian at the Canadian Wildlife Health Cooperative and the University of Montreal, and I'm grateful to present two posters today. The first one is all the steps that we do during a beluga health assessment. Most of you have seen what we do on the island, but there's a lot that happens behind the scenes, so we'll be explaining more of what we do when we get back to Montreal. And then there's also another poster on all the information that we can get from the reproductive tract. So how do we know if a female is pregnant, even if there is no baby? We can sometimes have information on the age of the animal, on depending, and a lot of other information that we can get. So if you're interested in hearing about those, I'm here on the side. Thank you.
Hi, I'm Ashley. I'm from DFO in Winnipeg. I do the aging for the whales in the ISR from all the communities, from the harvested samples. So I'm just presenting a bit about the different methods that we use to age whales, and a study that we did in 2021 to compare these three different methods. You can also come and see the average ages of the whales in the ISR over the past 10 years and the average age of the whales in the different communities from harvested samples.
So hi, I'm Laura Murray. I'm from DFO2, and my poster is actually by the door. It's on the Beluga Summit and just giving us some context and history on how we got here. Kind of similar to some of the welcoming talks, but just in poster form. So we've got like the origin that there was that we wanted more knowledge sharing, and then we had the 2016 Beluga Summit, and there's some information on the topics and some photos from that one that you can go check out. And then all the projects and programs that came from it, and then some information just on the diversity of the people here at this Beluga Summit, and then thinking forward to the ideas and programs that might originate from this Beluga Summit. So yeah, if you ever get overwhelmed with all the information, you can just go back to that poster and get orientated again, I guess, on what we're doing.
Thanks, Laura. Okay, coming across the room, Marianne.
Hello, I'm Marianne. I work with fisheries and oceans in Winnipeg. I have two posters. This first one shows the summer habitat selection of different groups, different group type of belugas. And this is based on an aerial survey that we did. And this one here looks at contact calls, so communication in belugas. And we compare different populations, so including the Eastern Beaufort Sea, but also St. Lawrence beluga and Hudson Bay beluga. And then we look at the different characteristics of their call. And what's neat is that the St. Lawrence belugas are in a very noisy environment, while the Beaufort Sea are very much quieter, and they do show difference in their calls to adapt to the different environment. Thank you.
Well, good late afternoon, or good late morning, almost afternoon. I'm Wendy Stott from DFO in Winnipeg, and my poster is just talking about an updated genetic analysis that we're undertaking, just updating some of the information that Leanne Postma had done in her great work a few years ago. And the poster is just about the methods that we're using and the geographic region that we're working on. So we're updating this information based on some ongoing work that is presented here today and some community observations. And what I'm sort of interested in is, as we get going on the analysis, just sort of any thoughts on questions that we should be, really looking into. What things are you observing? My poster has just an overview on the analysis we're doing and the places where we have collected samples to date that have come from the great archive that the DFO has, thanks to work from the monitors and folks like yourself who are collecting beluga.
Okay, thank you. I'm Keo, and we have two posters here from the FJMC. I'm gonna just try and walk away from the speaker. One in here and one by the door, and they're on the Beluga Monitoring Program and the Beluga Management Plan. They both have presentations tomorrow, so you'll hear more about it later, but they're a bit different from all the other posters because the FJMC is a management body, not a science body. So kind of taking the things that we're hearing yesterday and today and translating them into management actions and initiatives. And the Beluga Management Plan is a useful tool. You all got copies of it. So, and it's a useful tool that the FJMC tries to use in a more proactive way. So developing a plan in the ISR with contributions from the communities, IGC, IRC, and developing a plan here before a plan can be developed and imposed externally. And we're also developing a seal management plan right now, so come and ask us about it if you're interested.
Hi everyone, Kevin Scharfenberg. I'm a biologist at DFO in Winnipeg. I've got two posters. The first one is on the passive acoustic monitoring that we do in the TNMPA. So these are the moorings that we've been deploying for the past number of years with folks like Jimmy and Manny and Cody and probably others in the room. So this is looking at beluga vocalizations and sort of their beluga's distribution based on what we're hearing in the Delta. And then yesterday we heard a few comments about some of the bathymetry sonars and questions about the sounds that is introducing into the soundscape. So one of our master's students has been working on looking at how far that sound can travel in the TNMPA. So we've got some results on that, and happy to share those with people. And then the other poster, over the past couple of years, I've been fortunate to work with Kim Ovitz and the PhDC on the Pawatuck Beluga Drone Project. And in traveling to Pawatuck, we've heard a lot about skinny whales coming in. So we've got a bit of a comparison of body condition of belugas in the south versus the north in Darnley Bay.
Good morning, everyone. My name is Kim Ovitz. I'm a PhD student at the University of Manitoba, and I'm also with Fisheries and Oceans Canada in Winnipeg. I've been working in the ISR for the past 5 years with Lisa Loseto's team and the community of Pawatuck. So I'm here really representing this week the Pawatuck Drone Project. This is a partnership between the Hunters and Trappers Committee in Pawatuck and DFO with support from researchers at the University of Manitoba and NRCan. And we use drones and other technologies like hydrophones to look at beluga behavior and site use, and as Kevin mentioned, body condition in Darnley Bay. This is a really clear water area, so it's the perfect place to use drones, which is a non-invasive tool to look at beluga. And in this poster, we're talking more about behavior and site use, and we provide some examples of that at the bottom. Later today, if you come by and check out the table, we'll have some of our project gear with us and also videos, live videos the different things that we're seeing. So yeah, definitely come check it out if you're interested to learn more about drones, and also some of the work that we're doing with interviews and workshops to ensure that we're bringing Inuvialuit knowledge into these processes while we're interpreting and making sense of the things that we're seeing. Thank you.
Hi everyone, my name is Emma Sutherland. I'm one of Lisa's students based out of the University of Manitoba in Winnipeg. I have 3 posters to share today. So as many of you know, there was a tagging program in 2018 and 2019, which was a huge collaborative effort between a lot of people, and that would not have been possible without the support of all these different organizations and individuals in the ISR. This first poster here just gives a little bit of a comparison of the data that came out of that tagging campaign in comparison to the tagging data from previous years, just to show how detailed that data is in comparison to the older data. And that also shows us that the tags lasted for a very long time, especially in 2018. And you'll be hearing later from Luke about all of the cool questions that people were able to look at using that newer data. But a lot of the work that I've been doing is to try to bring together the data from all the different years, so from 2018 and 2019, alongside data from the 90s and the 2000s, to see if we could bring together all that information and look for changes within the population or in between years. And so I have one poster here that looks at the summer movements of eastern Beaufort Sea belugas. And one thing that we found is that when we pulled together all the whales from all the different years, there seems to be really distinct movement patterns that emerge. And so we can see that there are there's a group of large males that we see across all the different years from the 90s until today that travel directly north and do not come back to the Mackenzie Estuary after mid-July. And there's another group of whales that sort of seem to stay more within the Amundsen Gulf up until September, traveling back and forth in and out of the Mackenzie Estuary. And so that's really important as we look at things like aerial surveys or all these different programs, just to know that there's a big group of males that are traveling north and not coming back later in the summer. And then this poster here is looking at what these whales are doing after they leave their summer range, so during their fall migration, as they travel towards the Chukchi Sea and the Bering Sea. And so again, we brought together all this data from all these different years, and we saw that male belugas seem to follow several different routes, and they seem to be able to sort of mediate their movements in response to environmental change. So we saw, like in later years, as the sea ice retreated a little bit further north, these male belugas were able to travel into these ice-free areas a little bit further north and stop over all along these slopes north of Russia. Whereas in earlier years, when there was a bit more sea ice, they seemed to move a little bit more directly and quickly towards their winter range. So that's just good information to know about how these whales seem to be able to follow a bunch of different movement patterns as things are changing around them. Thank you.
Well, thank you, everyone. I know that a lot of work goes into these posters, and sometimes they don't get the exposure that they deserve. So I hope these little lightning talks have whetted everyone's appetite, and that you'll come and see them when we have the poster session later today, and check out some of the other props that are going to be around on the tables. So let's have another round of applause for all of our poster presenters. And if we could we have another round of applause for Chloe and Sonia, who I abruptly pushed off the podium. Very well done. Okay, so I'm looking over at Jen. Are we ready to be dismissed? Okay, so this brings us up to our lunchtime. We're actually slightly early, which is fantastic. We're going to reconvene at 1:30. 1:30, so you have a nice long lunch break here. We'll see you all back at 1:30. Thank you. Welcome back, everybody. I hope everyone had a nice lunch and bit of time to relax before we get back into the presentations. The next session is going to focus on distribution, abundance, and habitat use of beluga. And to set the stage, we've got Manny and Shannon here who are going to introduce the session.
Start? Okay. Good afternoon, everybody. Thanks for coming back. This is a great meeting so far. And we're really excited about this afternoon session. So today this afternoon, okay, that's better. We're going to be talking about what is a population, what is the size of a population, how is a population structured, for example, by size, sex, and age of individuals, where do animals move, what groups do they travel in, why do they go certain places at certain times, and what do they do when they're there. And so we'll be talking about that with a few presentations, as well as in some of the breakout groups later this afternoon.
As you know, we had a Beluga Summit in 2016, and we met with all the communities, and we had roundtable talks as well as what we're doing here. And pardon me? Oh, as you can see in the picture here, some of our Beluga monitors, some of our elders from the communities, we sat around a table and we discussed on the Beluga Summit. And you can see the late Fraser Pingo there, or not Fraser Pingo, Fraser Rogers. Sorry, I'm thinking of somebody else. Fraser Rogers, who was a Beluga monitor for here in Inuvik. And you see John, Danny, C, and all the other representatives from the communities.
And so just like at the first Beluga Summit, some of our goals today are to look at what do we know about Eastern Beaufort Sea belugas, and what don't we know that's a priority. And at the time of the first summit, we talked about the fact that the previous aerial survey flown to count whales and estimate the number of whales in the population at that time was 25 years old. That first survey was flown in 1992. So it had been a long time, and we really didn't have a good sense of the size of that population. And so we're going to hear from Marianne Marcoux later about this. And I think one of the main drivers for wanting to redo that aerial survey and abundance estimate was because of all the change that has happened in the Arctic marine ecosystem and all the things we've seen from a science perspective and that we were hearing from harvesters at the time. And so trying to get a better handle on the status of that population.
In the 2018-19 Beluga tagging program, previous tagging programs took place in the early 1990s and the mid-2000s. Change in the Arctic marine ecosystem since that time advanced in tagging technology, interest in collecting more recent beluga movement and dive data. And when back when they're in the 1993, 93, 95, when they did it, and when what's her name did it? Lois, Jack Orr. Jack Orr and Lois, when they did the tagging program back in the days, and they wanted the communities wanted to see where the belugas are traveling to, where they're wintering, and so stuff like that. And we wanted, it came up to the table again. People wanted to see where they're traveling to. Are they still going to the same wintering grounds and traveling? And that's why we did the beluga tagging again in 2018 and 19. And so was Alaska. They did the same thing in Alaska. They did tagging, and they put backpacks on them, and so that method they did, and so they're still going to the same place as when they traveled back in from 1993, 95. Yeah, and Emma's poster talks about that. And Emma's poster talks about that. So they're still doing the same thing. And I'll turn it back over to Shannon.
Thanks. And so we're talking about aerial surveys and tagging in this session, and we know that capturing and putting tags on animals is really a sensitive topic. And we've heard lots this morning about the long-term beluga monitoring program, and how much information we get from that collaboration to work with harvesters and sample the whales and have scientists look at those samples, but we can't answer all the questions using tissue collection from monitoring. And so aerial surveys and tagging are some of the other tools. When this proposal for these two programs first came out, myself and Lisa, we met with the Game Council over almost a 2-year period to talk about this work and talk about how we thought the program should look, how it should be governed, who should be involved on the project team, and then how we should reach out to communities about this. And so the Game Council asked us to go and visit all communities in the ISR. We met with HTCs and had open nights where we proposed the work, sought input from community members into how, what their research questions are, how we could design the studies, and answered a lot of questions related to concerns about tagging, and in some cases, aerial surveys, and also all the things that we thought we could learn from that work and how we would go about doing it together. So we went to the communities to talk about these programs before the work started, in the middle of the programs, and then again afterwards to report back, and that kind of ongoing communication was really important to helping us design our programs. One of the things that emerged from the first community meetings that we had back in 2017, and one of the things that I'm the most proud of and the most appreciative of was recognizing the need that the project team, the scientists on the project team, needed to meet more with our Nunavut partners to talk more about the details of these programs, because they were not ongoing work, and it needed a lot of discussion and thinking from different perspectives. And so we ended up forming a tagging advisory group. It included members from the Game Council, from the FJMC, really key support staff from the Joint Secretariat, and the scientists involved in the work. And we met sometimes every 2 weeks, sometimes monthly or longer, depending on what was happening. But we worked together from the beginning of these programs all the way through to the end. And I'd like to highlight that this tagging advisory group was really key in bringing people from the North Slope Borough on our community tours when we talked about our Beluga aerial survey, to also come and talk about their bowhead abundance survey. And that was a really great collaboration, and it was from the Nunavut members of the tagging advisory group that really pushed for that through our Nunavut and Inupiat Beluga Whale Commission. We did a lot of workshops together as well to make sure that we were really being transparent about our methods, especially for the tagging project, and to really hear from beluga harvesters what they thought about the work and how they thought we could design the work and do it different or better. The past tagging programs were from the early 1990s and mid-2000s, so technology has changed, and the ways that we know how to, say for example, disinfect versus sterilize our tags, there's a lot that has improved in our methods, and so we wanted to update the protocols that we use from a science perspective, and also bringing in a Nunavut knowledge, which really informed many elements of the field program, where we did the work, the timing, understanding whale behavior from a harvester's perspective to know how to minimize risks to them. There was so much here that we could talk about. And then of course, doing the work together in the field, we all know how important it is to spend time together on the land, getting to know each other and building those relationships and making it happen together. Should I keep going? So James Pokiak kind of mentioned this yesterday, but and I mentioned it in my poster. One of the things we really heard, and actually, Manny, I remember you saying about the tagging, we've been doing this for 30 years, there must be a better way or a different way. And we heard from communities that they wanted us to try to work on a way to tag whales that was based on traditional harpooning methods. And so we tried that in our first field season and demonstrated proof of concept, and came back together in winter 2019 and held a workshop. We were really lucky to have Melinda Holland, who was the CEO of the tag manufacturing company, join us, as well as a number of harvesters and members of our project team, and many of you are here in the room. And we worked together looking at the different tags, looking at people's harpoons that they make. We heard about people making their own harpoons yesterday. And then testing different tools on Muktuk, so working together on that. And I'm almost at the end here. So if you want to hear more about the harpoon tagging, please come and see my poster. I'll have some videos to show, and I think Luke is going to talk a little bit about some of the data that we got from that and the potential applications. So again, just to highlight how important it was to work together in developing projects that were new or hadn't happened in a really long time. I also want to introduce, because some people will see it on the agenda, that despite all of the successes that we had from tagging whales, and you'll see around the room the great data that we got from that, and we'll talk a little bit more about how that's informing the management of beluga whales to make sure there's a healthy population in the future. We did have some tagged whales die in one of the years, and it was a very surprising incident. It was really difficult for everybody involved. And when people heard about it, it was really upsetting for a lot of people. And we worked together to try to find out what happened and how to address that issue. So there'll be a talk at the end of this session about that, because I know there's a lot of interest in hearing what we found. So I'll stop there.
Yeah, and just to add on to in the incident there that we met in Yellowknife, and we had a big meeting there on. So that we met with pretty much all the group who was on the field team. We had Lisa, Shannon, and I was there as well, and pretty much everyone was there who was on the team and on the science and the workers. It happened not only in the ISR, it happened in Alaska, Eastern Arctic, and in the other countries too as well. Animals died off that year. I just wanted to mention that it not only happened here. I just wanted to say that a few times because it did happen all over the world, of animals dying and beaching up on the beaches. I just wanted to mention that they did everything in the right way. They did it the same as 2018. The times were even better at some taggings, right? Pretty much when they're putting on the backpacks and that, they were pretty much the same time. And just that year, we know stuff happens, not all over. So I just wanted to mention that and make it clear that we did have a big thing. It could have been a factor on the mortalities on anything. It could have been anything, weather, food, anything. So I just wanted to mention that.
Thank you. So you'll hear more about that from Emily and Lisa later, but I am so excited for you to hear from our next two presenters. So Marianne Marcoux is going to come up and talk about the aerial survey and abundance estimate for Beaufort Sea belugas. And then we're going to hear from Luke's story about so many amazing things that we learned from the tagging program.
Good afternoon. Thanks for coming back after lunch. We're sure that's going to work. I'm Marianne Marcoux with Fisheries and Oceans, and I'll be talking about the abundance estimate that we did for the beluga population in 2019. While this is searching, I have beautiful slides. It's fine. You'll see all my notes. That might be embarrassing. It's okay. There we go. Okay, I wanted to start by acknowledging my collaborators, Alexandra Mayette, Megan Ferguson, Claire Hornby, and Lisa Loseto. I just want to make sure. This? How do I go for it? Okay, and then it took a lot of people to do that survey, so there's many people to thank. And I'll talk about some of them during my presentation, but I want to make sure that I didn't forget that. So just to give you a bit of context about me, I work for Fisheries and Oceans. I'm based in Winnipeg. I'm also a mom. I find my happy place, it's similar to go hunting. I find my happy place going canoeing with my family, and I also like to beading. So as Shannon mentioned, there was the last aerial survey to estimate the abundance of the Eastern Beaufort Sea beluga population was in 1992. On this, I put the map to show what they did. So all the lines show where the plane was flying, and all the circles shows groups of beluga they found, and the size of the circle shows the size of the groups. So that was an amazing survey. It was led by Lois Harwood at Fisheries and Oceans. And this is the estimate that they came up with, about 40,000 belugas. But then, as was mentioned, there's been tagging studies to show that actually belugas were going way outside of what was surveyed in that last survey. So this is the study that Shannon was mentioning. So just the purple area show where the beluga spent some time. So they realized that actually the survey didn't count all the belugas, and it was probably an underestimate. We also heard at the Beluga Summit that it was one of the outcome of the summit that we needed to update this population abundance estimate. So that's when they asked me to do this work. So I was very lucky to get that job. So in the fall before the survey, we went on a tour with the 6 community in the ISR. And then we have representation from the Game Council, for FGMC, from Northborough, North Slopeborough. Just pointing out a few familiar faces. Jimmy was on the tour. John Oksana was on the tour. Gary was on the tour there, but you're not on the photo. And there's the late Craig George from Alaska that was with us. Yeah. So we visited all the communities and wanted to show the design for a survey to make sure that it made sense to local people, asking them for improvement to our design and also to when and how to do the survey. So this is what we came up for the design of the survey. So it's divided in different sections, and each of the black lines represent what we had planned to fly with our planes. So compared to the survey in 1992, we added quite a few new areas based on what people told us and based on the tagging data, so all the new. And also the pink area here, oh yeah, you can see it. Yeah, this was added after we did the tour that people told us that we should survey there because there's belugas. So the survey is kind of divided with the inshore and the offshore area, and I'll be talking a lot about the inshore and offshore, so it's just important that you understand that. Usually looks way cooler when you do animation. Now you see all my tricks. But okay, and this is the inshore area, just a little bit of a zoom section of what we are planning to do. One thing that we learned from a community tour, actually we knew already, but it was very important to have community members with us. So we were lucky enough to have Bertha Joe from Inuvik and Andrew Gordon Jr. from Inuvik, and Ron Inukta and Byron Okina from Hula Haktuk. So they were part of the survey as the same as everybody else in the team. So they did all the observation. I'll show you in a minute what I mean. So we're very thankful for that. One other thing I wanted to point out is that we did a training for egress, so basically getting out of a plane if it falls in the water. And then we did that at the pool just next door here, and it was important for us to bring instructor to Inuvik and provide this training to our local observers, and we also provided the training for other people that were interested at the time. So our survey had 2 different type of planes, so more traditional twin otters on the left, and a remotely piloted aerial system. So these are basically fancy drones. I'll call them airpass for the rest of the talk, and I'll talk about those 2 different aircraft for the next few slides. So So in the Twin Otters, we had 4 survey observers. So typically it would be 2 DFO staff and 2 local observers. And then they're there to count belugas. The planes have special windows that you can see on the left. So you can really put your heads in and really count the belugas. So that's quite important for survey design. And we use those planes to do the offshore areas. And then we flew at about 1,000 feet or 300 meters. And then we used the RPAS to fly the inshore areas. And the RPAS was equipped with a camera that would take photos. Obviously, there was nobody in it. And we expected to have higher densities of belugas in the inshore areas. And we know that observer can get overwhelmed when there's too many belugas, so we thought that would be a good method to do the photos in the inshore area. Just if you're curious, this is a pretty big plane. It took, you see all the people in the yellow vest, it took like 6 pilots, engineers to make it run. It takes a proper runway, so it worked nicely in Inuvik here. So it's not something that you can just improvise, like it was quite a bit of work to do. So each photo in the RPAS was georeferenced, so there was a GPS point with each photo. So on this figure, there's a bunch of little rectangles, so these represent all the photos that were taken, so then we can kind of glue them all together. And then we had someone who went through all the photos manually and counted the belugas in there. Yeah. And for the people in the front, I'm not sure in the back, there's little red dots. That's where they detected belugas in the photo. This is what the photos look like. There was quite a bit of discussion yesterday about clear water and murky water, and I'll talk a bit more about this, but you can see an example of this. And then when you zoom in, that's what the belugas look like in those photos. So obviously, when we fly the survey, we can only see belugas at the surface of the water, but we know that belugas dive, so we need to correct, to adjust our survey for the belugas that are underwater. So this was the first time that we could develop an adjustment factor just for the Beaufort Sea belugas, and this is thanks to the tagging program. I'm not going to talk much about tag because Luke's going to talk about them, but I need to tell you a few things. So those tags provide the location of the whale, so that was important, and they also provide some depth information, some diving information. So first, we could overlay the track of those whales with the survey area, and I remember I mentioned the inshore and offshore, so we could develop some adjustments that were specific from the inshore and specific for the offshore, because we know that beluga behave differently when it's quite deep or when it's quite shallow. We also know that the water clarity is different in the inshore and the offshore. So that was important to know, for us to know where the whale was and develop 2 different correction factors. So this is just to give you an idea what the data looks like. So at the bottom it says like from 8 p.m. to 10 p.m. So this is just a time snap in the life of one of the belugas. So at the top where there's a little wavy line, that's the surface, and then you can see the dive profile, so the going up and down, that's the beluga diving, and I just zoomed into the surface because that was interesting to us. So for the clear offshore water, there's been some experiment, and we think that we can see whales up to 5-meter depth. So what we did is that we calculate how much time whales spend in the top 5 meters, and then we can calculate. So that's what I illustrated here with the gray zones. And then we calculated that they spend about 50% of their time at the surface and 50% of their time at the underwater. So basically we can only see 50% of the whale. So at the end, we kind of need to multiply our estimate by 2. And then we derive another estimate for the inshore based on the top 1 meter. And they spend about 64% of their time at the top of the water. Okay, some results, and I have a few minutes left. So remember my first map where I show what we wanted to do. So this is the map of what we actually did. Maybe we got a little bit ambitious and excited, but the polygons that are empty are the section we didn't do, and the polygons with colors are the section that we did. So on this map, you can see the line where the plane went, and this is from the Twin Otter with the visual observers. And each of the circle represent a group of beluga that was seen by the observers, and the size of the group, the size of the circle represent the size of the group, so from 1 beluga to up to 5, 6, 7 in a group. And then this is a similar map, but this is from the inshore, and this is what was taken by the photographic survey. And on this one, each circle just represents 1 beluga that was detected in the photos. So the big scoop is that the number haven't really changed. So we came up with an estimate of about 38,500 belugas, rounded to the 100. And so it's not too different from what was calculated from the 1992 survey. One thing is that we did cover a little bit larger area. So on this map, the solid color is what we covered, and the hashed ones are what was covered in 92. So you can see that we did cover a little bit larger area. But again, I don't want to steal too many scoops from Luke, but the belugas that were tagged that year were going all the way outside the area that we surveyed. So this is probably an underestimate of the abundance. And I've been told to wrap up, but I just want to point out that there's a poster in the corner that Alexandra Mayette did. And just to point out that there's lots of money that go into the survey, but the data can be used for other things. So she was looking at environmental variable and to see where belugas were and trying to understand what kind of habitat beluga like. So there's way more things we can do with this data. And I'm done, just to re-thank the people that help us. Thank you.
Thanks a lot, Marianne. That's really impressive. And nice to see that the population is still healthy. Do we have any questions for Marianne? No? Okay. There will be opportunities later. So then in that case, what we'll do is we'll bring up Luke. And I think Marianne has sort of whetted everyone's appetite. And so now we're going to hear the details of the work that Luke's been doing on the beluga movement and dive behavior.
Okay, so hi everyone. Thank you so much for having me here today. It's really great to be back in Inuvik again. I was here 6 years ago when we first tagged beluga whales, and I've spent a lot of the last 6 years analysing the data that we got from these animals. So I've been looking at their movements, their dive behaviour, looking a bit at their foraging, which habitats they're using, and we've also looked at their interactions with shipping too. So I'm really excited to share some of the results that we found with you today. So as Shannon mentioned, belugas were tagged back in the 90s and in the early 2000s, but given recent signs of ecosystem change and potential threats to beluga whales, there was renewed interest in updating our knowledge on their movements and their dive behaviour. So between 2016 and 2019, a number of meetings took place between scientists, community members, and Inuvialuit organisations to discuss what we wanted to find out about these whales and how we could do this. And these meetings included the first Beluga Summit too. So a number of themes emerged from these meetings. People wanted to find out what they're doing throughout the year, so looking at their seasonal behaviours and movements, not just in the summer, but finding out what they're doing in the winter as well. They wanted to look at their migrations, update our knowledge on their dive behaviour, and there were also concerns about the increased levels of ship traffic and how this could impact whales. So in 2018 and 2019, we tagged belugas again, and my PhD was all focused on analysing the data from these 2 years. So we tagged whales from Hendrickson Island in Cook-Mallet Bay, and we used 2 methods to tag whales. Firstly, we used this live capture method, and this is how belugas have always been tagged in the past throughout the Arctic, so in Norway, Alaska, and Canada.
And this involves capturing the whale, holding it in place for some time, and attaching this tag to its back. We also, as Shannon mentioned, developed this harpoon method. It was co-developed between Inuvialuit knowledge holders and scientists, whereby a whale is followed and a tag is deployed on this free-swimming animal. So again, if you want to find more about that, go and see Shannon and Dennis at the posters behind me later. So these tags collect a lot of data. For my PhD, I analysed data from 16 of these whales. So all of the whales that we tagged were males. We did try and tag females, but we ended up kind of accidentally always tagging males, so they're all large adult males. The tags tell us where the animals are. They collect location data, so we can track them over time. Most of these tags transmit data for a few months, but we had some tags that transmitted, told us where the animals are, provided data for about a year. They collect depth data, so they tell us how deep the animal's diving, how long it's diving for, and we can also look at the structures of their dives to get some sense of what behaviors are represented by individual dives. And some of these tags also collect temperature data, so we can get a sense of the environmental conditions that the animals are occupying, what they're experiencing. So together, this data allows us to look at the movements and dive behavior of belugas, as well as the environmental conditions that they encountered over an annual cycle. So far, we've published 7 papers using these data sets just from 2018 and 2019. You can access all of these papers online. They're all open access, and I'll show you them again at the end if you want to take a photo and get the titles. You can just Google them and find them all online. But we've looked at their feeding behavior, we've looked at their movement behavior in relation to the environment, we've looked closely at their migrations, their responses to ships, and also their habitat use and hotspots. So I'm going to talk about some of our key findings through the story of a year in the life of a male beluga whale. And before I show you that, I'm going to show you an animation, which is an animation of all of the tracks of the whales that we tagged in these 2 years. So I've combined the 2 years together, so you're going to see whales from 2018 and 2019. So we can see that when we've tagged them, they leave Cook-Mallet Bay and head to these deeper waters in the Arctic archipelago, so Amundsen Gulf and Viscount Melville Sound. And then when we get to late summer and fall, they migrate westward over the Arctic basin. These are really deep waters, around 3,000 meters. In October and November, they start to move into the Chukchi Sea, and we see these animals coming a lot closer together again after having been apart for several months. They move into the Bering Sea over winter, and they spend several months there when the Bering Strait is blocked by ice. Now, of course, it's hung up. Oh. And now it started again. I do have some more animations that kind of show the rest. But basically, then the whales migrate northwards into the Beaufort Sea in spring. And most of the last few tags stop transmitting in June, so just before we tag whales in July. So I'm going to talk about a year in the life of a male beluga to demonstrate some of the patterns that we found in these different papers that we've published. So in each of these slides, you'll see a map in the top right corner that shows America, Canada, Russia, and in each of these maps, I've highlighted the approximate area where we see belugas in each season. So our story starts with a tagged male beluga that's leaving Hendrickson Island and is about to head out into Cook-Mallet Bay. So when that whale leaves Cook-Mallet Bay, we know it's going to head off to Amundsen Gulf to the east, or it's going to head north into Viscount Melville Sound. And we know that this is an important time of year for foraging behavior by whales. They seem to put on weight at this time. And when we look at their dive behavior, so this figure in the bottom right shows a series of dives by a whale. So you're seeing the depth that's reached by a whale over time. And in this specific example, I showed dives over a 24-hour period. So you're seeing the whale swimming to the seafloor at around 450 meters, coming to the surface again, and then repeating this behavior. So you can see the seafloor at the bottom in gray. And we find that at this time of year, belugas are making these really deep dives to the seafloor, consistently to around 400 or 500 meters. So in this particular example, the whale made 50 dives to the seafloor over a 24-hour period. So even just vertically, it means that this animal is traveling 50 kilometers in the day because it's going to 500 meters and then to the surface again. So these dives are likely quite costly for the whale to make. They're very deep, they're very long. But from the few dietary studies that have looked at this, and also from our knowledge on prey distribution, it seems like whales are likely targeting adult Arctic cod at the seafloor at this time. It's a great prey species for them. So some of the whales, although most of these whales seem to stay in these deep offshore areas, we find that some of these whales move back and forth between the estuaries, between the coasts at this time. We don't know exactly why this is, but in 2019, Colin Gallagher and his team tagged a Dolly Varden, and this Dolly Varden was eaten by a marine mammal. We know this because it was recording cold temperatures. We knew the fish was in water, 3 degrees Celsius, 3 degrees Celsius. All of a sudden, the tag's recording temperatures of 30 degrees Celsius. It's in the stomach of a mammal. So we identified that this mammal was likely a beluga, and we've looked at the beluga track data alongside this to see that belugas do often move forth between these estuaries. So we don't know exactly why they do this, but there's a possibility that some of the whales return to these coastal environments to feed on fish that are migrating from the rivers in the summer. We looked at what kind of environmental conditions belugas forage in, and we found that they feed over a wide range of sea ice conditions. So we see them feeding in areas with really dense pack ice, but also open water. But they seem to primarily forage in areas that are over a narrow depth range, so around 350 to 700 meters. So we can use this information to model and then map their foraging habitat suitability. So on this map, the warmer colors denote the more suitable summer foraging habitats. So you can see that the most suitable habitats occur in these deep channels in Viscount Melville Sound, in the Amundsen Gulf, and somewhat along the Beaufort Slope as well. So we know that these areas are likely to experience increases in vessel traffic in the near future as the sea ice coverage declines. So we hope to use the information from these studies to help inform conservation measures to help protect belugas from the impacts of increased underwater noise from these vessels. We also used the beluga tagging data and matched it up with, oh, I need to play this. In fact, I'll play it in a second. We matched the beluga tagging data up to vessel data to see how beluga whales respond in terms of their movements and their dive behavior to a ship passing nearby. And this study was led by Morgan Martin and Bill Halliday at WCS Canada. And so in this animation, you see a beluga heading eastward, and then it stops for a while. It's likely starts foraging. And then we see a ship coming from the east, and immediately the beluga moves away. And this is in Amundsen Gulf, in the kind of middle of Amundsen Gulf that this occurred. So Morgan found that there were signs that belugas changed their movements, changed their dive behavior, potentially they were disrupted in terms of their foraging behavior by ships coming nearby. Okay, now we get towards the end of summer, and so we're in late August, early September. Belugas begin to migrate westward over these deep waters of the Arctic basin. So now they're going into waters that are a few thousand meters deep. So this phase in the beluga movement cycle is often called a migration. We see them making these really long distance movements between a distinct summer area and a winter area. When we zoom in on these tracks, again, that's what we'd expect from a migrating animal. They're moving in a fairly straight line. They're moving around 100 kilometers a day. But when we look at their dive data, so you see in the bottom figures here, we see that they're regularly diving to 600 meters or 800 meters even during these long distance movements. So this isn't really what we'd expect. It looks like the whales are constantly searching their environment. They're potentially even foraging during these long distance movements. This is very different when we compare it to the likes of humpback whales or blue whales that undertake these long migrations between high latitudes and low latitudes, and they do so without feeding. Belugas seem to be feeding during these movements. So this is what we'd call more nomadic behavior, and there's signs that belugas are constantly searching for their environment, potentially learning about it and responding to these resources. And nomadic behavior, this kind of ability to respond to the environment as you find it and learn about it, is an adaptive behavior to exploit unpredictable environments. So this kind of behavior might be useful for belugas to adapt in the Arctic, which is obviously experiencing unpredictable changes in wind, ice, sea temperature, and so forth. So Emma has some posters on beluga migrations as well. Emma Sutherland, she talks about the different migration routes that are used and also the summer distribution among different groups of whales. So if you want to go and see her posters, go and see them on the other side of the board and talk to Emma later. We see something else interesting happen in fall, during these fall movements. So when belugas start their fall migration, they experience 24-hour daylight. The sun never sets. And at that time, if you focus on this top panel of figures now, we see that whales don't have daily behavioral cycles. They do things at random times. They feed at random times, they rest at random times. But when the sun starts to set, later on in fall, you have these distinct phases of day and night. And when we have that, we see these belugas start to develop really distinct behavioral patterns. So in this bottom figure, you can see that during daylight hours, at the far left of this figure, they're making their deepest foraging dives to around 600 meters. During the night, they're making these shallower foraging dives, about 200 meters. And then in the morning, they seem to be resting or potentially traveling as well. So these slides were probably easier to follow when things appeared, so I apologize for that. But we also found that they changed their behavioral cycles depending on other environmental conditions. In shallow areas, even when belugas experienced day and night, they behaved randomly, basically. They fed at random times, they rested at random times. And in these really deep areas, they seem to depend, these behavioral patterns depended on the thermal structure of the water column. So when you had distinct layers of warm water and cold water, the whales seem to have these patterns that suggest that they're using their dives to track prey that's migrating vertically over a daily cycle. So it seems like they're following fish that's moving between this warm water layer and the surface between day and night. So this is something we've only just looked at. I thought it'd be fun to make a new animation. But throughout fall, we see that belugas are fairly far apart from each other. These tagged whales a few hundred kilometers apart. When we get into October, they enter the Chukchi Sea, and we see these animals come really close together. You can look at this animation here and see that these dots of individual whales overlap at times for a couple of days at a time. I'll let it play again just so you can see it all, but it does look like there's times when there's 2 or 3 of these tagged whales that are potentially sticking together. It's paused. There's around 3 whales there that seem to be staying very near each other and potentially traveling together for a few days. So again, we haven't analysed this in detail, but there's potential signs here that after having a month or 2 apart, these whales are coming together, and it's an important time of year for social activities, some different social behaviour, maybe even sharing of knowledge. So when we get into November, the belugas start to move southward into the Chukchi Sea, and then they into the Bering Sea, and then they spend several months there as the Bering Strait is blocked by ice. So when we looked at the dive data for belugas at this time, we were surprised to see that they're really diving intensively to the sea floor. So the Bering Sea is pretty shallow, around 50 meters, and we see that belugas are pretty much always making these dives to around 50 meters. So in this figure here, you see a series of dives over 9 hours. You can see the whale keeps going to the sea floor and then coming to the surface again, and it's spending very little time at the surface. There haven't been many studies on the diet of whales at this time, but they likely feed on a wide range of different fishes and invertebrates. So this is something we've only just started looking at as well. When we're getting towards the end of winter, we see that whales really seem to be sticking close to this ice edge. So this animation shows February to April. And we can see that the brighter white pixels denote higher ice concentration. So this animation's on a loop, so you should see it again. But it looks like the whales are staying close to the ice edge, potentially waiting for it to break up before they can move into the Beaufort Sea. But what we see here is 2 of these whales that transmitted data throughout the winter. I've stopped again. I'll just go back, let you see it again. 2 of these whales seem to migrate a lot earlier, about 3 weeks earlier than 1. So they're migrating north when there's still a lot of ice there. They must be going through quite narrow leads and cracks. Yeah, you can see one head off there, and then a second one follows. And then this third one waits about 3 weeks before the water's opened up a bit more and then heads through. So then when we get to spring now, we're in April through June, we see the whales migrate along the Beaufort Slope. They tend to stick along those deep waters that are around, well, it goes from 500 meters to a few thousand meters deep. And at this time of year, we see whales making their deepest dives. They're regularly going to around 1,000 meters. And we even recorded the deepest dive ever recorded by a beluga in this study, which was 1,400 meters. The studies on prey in this region suggest that they're likely targeting Greenland halibut and Arctic cod. So that takes us towards the end of our year in the life of a male beluga, and we can see that belugas re-enter the Beaufort Sea in May. And when they do this, they pass right by these coastal regions. We don't see them coming near the coast in May and June. Instead, they're staying in these deep waters of Amundsen Gulf or foraging along the Beaufort Slope. And there's one whale that heads further north past Viscount Melville Sound. So this isn't the best quality ice data, but you can see that there's some ice blocking these coastal regions at this time. But it doesn't appear like the whales are going near the ice to check it out. Instead, they're staying in these offshore areas, continually foraging. So just to summarize, we've looked at the animal movements, their dive behavior, their relationship with the environment, and we've done this over an annual cycle. And it's allowed us to look at their foraging behavior, their habitat use, interactions with vessels, and also seasonal changes in behavior and migrations. One of the things that I take away from this collection of studies that we've done is that eastern Beaufort Sea belugas exhibit real flexibility and diversity in their movement strategies. We see that they can feed at 50 meters or the surface or 1,000 meters. They can feed at the seafloor or in the middle of the water column. And they seem to be able to adapt their behavior depending on environmental conditions and likely prey assemblage. They can forage over a wide range of ice conditions. And even most of the year, belugas don't have these daily patterns in their behavior. They do things at random times. But under very specific situations of light, water column temperature, and seafloor depth, they have these distinct daily patterns in their behavior, which suggests that they're able to target prey, which is migrating vertically. And they can do so because they don't need sight to see their prey. They can echolocate, they can find prey in the dark. And they have these complex long-distance movement strategies that suggest that they're constantly searching their environment, potentially learning about their environment too. There's still a number of unanswered questions. For example, we know that a number of whales are often seen in Darnley Bay, but none of the tagged whales have ever been there. So potentially there's patterns that might be shared among whales that are related or are part of the same social group, which we haven't been able to test yet. And also this study, everything I've talked about so far is all based on males. We still have a lot to learn about females and juveniles. I think that one of the methods that could help us answer some of these questions is this remote deployment method or this harpoon tagging method. Because this method allows us to follow, or Inuvialuit knowledge holders to follow a whale and tag a free swimming whale. So we can tag multiple whales that are found in the same group, potentially take tissue samples from these animals as well to find out whether they're related. And then we can test whether they travel together, whether they share movement patterns. So in this animation on the left, you can see the tracks of 3 whales that were tagged on the same day in 2019 using this harpoon method. And they share some similarities in their movement patterns. They both head west before heading north, and 2 of them stick together briefly for a few days. So as Shannon mentioned, her and Dennis have a poster on this, and it'd be great to talk more with them about this. So I want to just wrap up by saying thank you to everyone who's been involved in helping me analyze this data, but I want to give a special thank you to everyone who's been involved in this project, whether you were in the field with us or helping with logistics on the ground, working back in Winnipeg at DFO. There's been so many people who've been involved in this project, and I've worked with a lot of different whale data sets now, and I can honestly say that this is one of the most comprehensive and complete data sets I've ever seen. It's really impressive the amount of questions that we've been able to answer with this data, and I hope I've given you a glimpse of that, and I wouldn't have been able to do it without having such an amazing team of people at every stage of the process. So thank you. And I will just finish by also acknowledging and thanking all the partners and funders. And I'll leave this slide up now, but you can access any of the publications freely online. And we will make these animations, all of the animations I've shown, available on a Beluga website, and we'll let you know when they become available. So thank you very much for listening. I'll welcome any questions.
Thanks a lot, Luke. That was absolutely fascinating. Do we have any questions from the audience? Okay. All right, I'll start in the middle of the room here.
What's the farthest east one of the whales has gone?
You can just about see it in the track there. It is as far into Viscount Melville Sound as they could go, and it's I'm pretty sure it's blocked by quite a lot of land-fast ice when they get there. So we know that there's another beluga population just to the east of that, but it hasn't been mixing between them yet.
Hello, Warren from Saks. I was just going to ask the same question because of me and my son, we watch lots of National Geographic, and you see the other pod of whales that goes to Elefringus, where they peel their skin. Do you ever, or in the future, are they going to try and tag a female whale to see if they go up there?
That's a good question. We don't see any of the tag belugas from this population. They don't seem to go into very shallow coastal areas where animals typically molt. We think, anyway, that's the main reason to go there. None of these tagged whales go in those environments after they leave Cook Mallet Bay. Well, sorry, some of them do pop back to these areas along the Beaufort Sea, but certainly not in the archipelago, the Arctic archipelago region.
Yeah, this is the only reason I was asking, because all those are males, and like you see on National Geographic, they'll go to Elefringus to go and molt. And you said, or that's just my question, is if you guys in the future will try to tag a female whale.
Yeah, like I said, we did hope to tag some more females when we went out, but it just so happened that it was mainly males that we tagged. But I think this remote deployment method could give an option to potentially tag more females in the future. So it would be nice, but there's currently no specific plans. But
any other questions?
I'm trying to think of the other two places where they've been. Can someone remind me where they were tagged in the 90s and 2000s? It was all. Say again, sorry. Kendall Island, yeah, they were tagged there in the 90s and 2000s, early 2000s.
Marianne, and then Jerry, and the gentleman here.
Thank you. Looked fascinating. The whales that were reunited in the fall together, were they tagged all the same day or around the same time?
I don't know actually. Admittedly, this part of the analysis is something I think we should look more into. I've literally only just done that within the last couple of weeks. I thought it'd be neat to see, do whales meet up? But they would have all been tagged over that same couple of week period that we tagged whales, so 2 weeks. But yeah, good point. I'll have a look at that.
I got a question. I was watching how deep the whales dive. 1,500. That's about 4,000 feet. Those tagging, like the harness and that, can they hold the pressure when they're down there? Like they won't be crushed like like that submarine for Titanic?
Yeah, they're pretty impressive, these tags now. They can survive around 2,000 meters. Similar tags are used on sperm whales, which go to over 2,000 meters. So they're pretty hardy.
Dennis Airy from Macquarie. The tagging, they were sort of selected when we were deploying the tag. If we see females with young ones, we just get away from the pod. Because the female, at that time of the year, they're giving birth, and on the long migration, the okra ain't that thick on it. So it was sort of a safety thing. So the dart don't go into the meat. And through traditional knowledge, the elders tell us that different whales go different places. If you see those tagged whales, they never went in towards Polituck where they hunt whale. They didn't go towards the west side where we hunt whale. You know, it's through TK, the elders say different whales go different places. And we see it in September when they migrate from Shingle. When we're down there for miles, you could just see nothing but whales going west. I think one of the regulations that tagging it was, you know, with the advisory group, one of the recommendations come out is try to avoid females. Because you tag a female and you injure it and they got a cap, and beside after they give birth, they lose quite a bit of the fat. And
Yeah, thanks, Dennis. It's great to see you again. That's a great point about the females. Sorry I didn't make that clearer.
Thank you, Luke. Amazing presentation. Herb from FGMC. I was just, in my other role, I was just at the Central Arctic Ocean Fisheries Agreement in Iceland last week, and each of the countries gave a presentation just like yours of all the species of fish that they're catching around the Central Arctic Ocean, at least on the 200-mile mark. In our region, and USA, Canada, and Russia's regions are very similar, like a lot of Arctic cod, a lot of pollock, and a lot of flounders. So it's maybe, and at certain times of the year, and we're thinking that maybe that might be in the fall, where they're getting all the beluga or all of your samples or what they're fishing. So all those sailings happen, I think, after August 3rd, when pretty much the Northwest Passage opened in that area. So that might help put a picture together of the region that's so vague with all of the research, being disconnected with the countries, especially the way they are now. It's a really good collaboration and coordination. It's the first time we've ever seen data from Russia and what they've been sharing with us as a group after about 10 years. So it's good to see. And this really this is probably the most detailed story I've ever seen of any research in the region. Thank you.
Thanks, Herb.
Yeah, thanks, Luke. I'm just wondering, I just watched Marianne's presentation there about the whale population and stuff like that, and then, we heard over yesterday and day before yesterday, in the 70s, 80s, we'd be taking over 200 whales in our ISR area and that, and now between all 6 communities, maybe we'll get 70 or if that from all the 6 communities, and then with Marianne's data there, it's lower than in the 92s and that. So, and we're harvesting a lot less beluga whales than that, and then through all knowledge in that, you'd think the whale population would have probably doubled, and instead of got a little bit lower, and they're still saying it's a nice population, but you'd think it'd be a lot higher percentage of whales, and I'm thinking maybe because the ocean is opening up so much, the whales are going in all different directions, like your data are showing that, so they're not all staying in one big groups of pods and that. Now they're kind of going in different directions where a lot of whales could be missed. And so I'm just wondering, with this tagging data, along with Marianne's work, you could probably get a better estimate of the beluga populations in the whole area.
That's a really good point, Jim, and that's one of the things that Emma Sutherland is working a bit on, looking at these different summer movement patterns and migration patterns, and looking a bit how they're related to the environment. And we do see a few discrete groups of we do see a few discrete movement patterns that are present across years, but there's different numbers of whales that take these. So there do seem to be some. So maybe whales are going to different places over different years.
Yeah, because in saying that, we know we have very good research on all the beluga whales and stuff like that, and then it wasn't mentioned yet or anything like that, but we know through research and age of the whales and that, beluga whales lives over 100 years old and that, so those whales got to be still somewhere out there because we weren't harvesting as much in that, and even Alaska side is not harvesting as much as they used to harvest back then.
Manny?
just as a harvester, when we go out, like some years you'll see lots of belugas when you go out, and after you finish harvesting a beluga and you're towing back in, and you'll see whales all over when you're towing back in on the west side. And that's where I hunt, and some years you see when after you finish harvesting a whale and then you're towing back in, you'll see whales all over when you're towing. And then some years it's different because we're going 20, 30 kilometers out to harvest belugas where we go to shallow water, and it's murky water, and some years is next to nothing. Like you'll go out, and you make a big circle to where we always hunt, and we have to go a long ways out when we hunt, and it's like I said, some years is good. You'll see when you're towing back home, hundreds of whales all over, and then some, like last year, like it was a tough year, it was, we had to go way out, and coming home, you'll see maybe 5 or 6 whales at the most when you're towing in. And last year with whales, there's big bulls that we seen, and like we don't hunt when we see a pod of whales, we'll watch them come up and make sure there's no young ones. If they got young ones, we'll leave them, go look for another pod of whales, and that's what we do. And last year there was that's the biggest whale I ever gotten, as a captain, and was 14.7, and it was just 10 feet around that when they measured it. And so, some years you'll see, and then me and my son-in-law, we hunt, Kimmy goes out with another boat and I go out with one boat, and we'll travel out together, and you'll see the different hooks of, the one we got, ours was maybe 5 inches thick, and theirs was 3 inches, totally different, their whale was skinny, I don't know what they're both males or, yeah, both males too, and you see stuff like that, and you see old wounds on caribous that what we take and we send for sampling, and I just want to tell one story. Well, I know I got 2 minutes. Last year, years ago, I hunted with the late Dennis Hansen and Alec Avigana, and I went out with them from Shingle Point, or from on the west mainland side, and we went out, and I was a young boy. We went out, and they're both experienced hunters, they're elders and they know, and they went out with Alec Avigana's big boat, and there's some tough whales, and the whale that we chased, we started chasing it in Shallow Bay, and it never died. They shot it. I don't know how many boxes they used, but man, there's some tough whales. They lost it, we lost it in the deep water, and it was gone. It was hit how many times, and we tried to get close to harpoon it, but that whale was a tough whale. There's some tough whales out there that doesn't want to give up dying, and I don't know wherever it had to die somewhere in the deep water because it was shot so many times. You lose whales like that. You lose whales they sometimes you get some hunters that you shoot them in the head, they sink right away, and people go through that. There's whales that are lost. You just mark it when you come in, and you say, I lost a whale. I mean, it is what it is. I mean, you can't do nothing about it. It's sunk, and so just things like that. There's some really tough whales that doesn't want to die. I know I heard it from my dad. Aggressive whales. I heard it from someone told that story of aggressive whales at Georgia, I think it was, that told that there's tough whales. There's whales that are aggressive, And when they don't want to be hunted, they'll turn around on you and hit your boat. And I heard that from Georgia. I heard it from my dad. And whales that, like I said, we chased it and it never died. It was a big male too. And so stuff like that, when you hear stories, and a really good presentation you gave, I thank you for that. And it's a really good presentation of the belugas.
James?
Thank you. Just wondering, I know I've worked with Fish and Game in Alaska in the past, and I know they also tag belugas, bowhead whales, and we've done that in the Beaufort Sea. Is there any way to make some kind of a comparison to the different pods, the different areas that they migrate and spend the summers in? So I'm just curious if you guys know if there's any difference in the diving patterns of the Alaskan side versus the Beaufort Sea.
Yeah, it's a good question. They've over the years they've undertaken similar analysis to what we've done, looked at their movements across the year and dive behaviour, and we find that there's a different population that they tag. They tend to go into estuaries closer to Alaska at a different time, and they tag that population. The whales that they tag don't go as far east into the Arctic archipelago. They tend to stay, if I remember the tracks correctly, spend a lot of time in that deep central Arctic basin area and also off Alaska in Barrow Canyon. I do recall in those papers those whales made fairly deep dives as well. I think I saw 300, 400, 500 meters. I don't know if they used the same tags that were able to record individual dives. The advantage of some of these tags were we got this information of individual dives, so we could say, yeah, that dive was 1,000 meters deep. They got some more summarized information from what I've seen published anyway. And we also know that these groups of whales occupy slightly different areas in winter too.
Yeah, Luke, a couple of years ago I was working with the bathymetry program, and I got a guy to come with me in my boat just to have somebody else in my boat. And he told me way back, I don't know how many years ago, but there used to be a different pod of whales, like a big, on the north side of Hendrickson Island, they were big whales. They were so big that they never went into the estuary. I don't know if anybody here, maybe Lenny or my dad or somebody could speak to it, but they were bigger whales, way bigger, way back in the day before outboard motors and all that good stuff, before we had big boats. But Noel Raymond told me that story. He said the old timers used to go on the north side and get a big whale if they wanted a big whale. And those were 20 plus whales, he said. Thank you.
I got another question. I mean, this is like with the storm surges that we're having now, and this came up, we're seeing more belugas that come into the delta. And a few years ago, about 50 or 60 belugas came up, and those are all big males. And they're a clavic harvested 11, I think, and like they were 16 footers, 16 plus footer, 15 footers. They were all big whales that they brought to the community of Aklavik, and they cut them up there like they were all. And some we're starting to see that trend where they're coming into the, like as far as someone told a story, and as far as McPherson, that was a young whale that went up there. And so I mean, and then you're hearing, like we're seeing whales that come by Aklavik. More, I mean, we're starting to see it in like more now, because these big storms that they're having, maybe that's a big storms that's pushing them into the delta. And someone said like there's different pods of whales, like them whales that they tagged, they never went over to the west side or to Politek side. And so this stuff like that, it's good to hear stuff like that, and it's good to like note that down, that, you know, like if they ever do a tag in the future again, that they come and do, you know, like one or two in the west, yeah, the Politek and Nuvik and tuck, and they're going to be a really good thing if they ever do. So,
I think that's a great point. It's great to hear from you and John and Dennis and all of you about these different potential movements of whales that we haven't seen in the tagging data. You know, one of the problems with tagging is we get a small number of whales, so there's potentially way more movement strategies out there. Some whales, as Dennis is always saying, some whales to go different places, different whales go different places. So we're probably only capturing a fraction of this diversity in the strategies, maybe.
I guess two questions. One, has there ever been a tagging program of the polytech whales? And two, is there conclusive data that they're the same population?
I'm not sure if there has been tagging polytech. Well, no, there has not. No, the first one. And I'm pretty sure genetic studies suggest that they're the same population. Yeah. Thanks.
All right. Well, that was a fantastic discussion. I really want to thank everyone for their input. James, did you want to say one more thing? Okay.
I'm just curious. A few years back, I was involved with the Alaska Fish and Game and DFO and tagging bowhead whales. Do you all know that there was an actual gray whale that was tagged in the Beaufort Sea? That one year we were out there and we accidentally put a tag on a gray whale. So I'm just curious.
I don't think I did know that actually. Do you know if did they track it for a while? Do you know if they? Okay. Thanks for that. Yeah, very cool. I'll have a look into that.
Interesting. Okay, thanks for that, James, and thank you everyone for all your questions and comments and sharing your knowledge. And thank you, Luke, for... Jerry? One more. Okay, last one. You know what? This is why we're here, though. Down.
James, you brought up a gray whale. A couple of years ago, Davy and Ruthie, they seen gray whales right outside of whitefish. They couldn't figure out what they were. They were black, and then they ended up telling them they were sub-adult gray whales. That's right in front of whitefish. Just information.
Thanks, Jerry. How about a big round of applause for Luke? So as we heard in the introduction, despite everyone's best efforts, despite employing all of the best methods, we did have some tagged whales die. And the team, Lisa and the team and the knowledge holders spent years looking at that event, and that culminated with a meeting in Yellowknife that Manny referred to. And so now, to end off the session, we're going to hear a presentation on the findings of that investigation.
Just as we get started here, I did want to, there was several times mentioned the idea of different groups, different pods, observations of why none of the tagged whales went into Polytech. I did want to highlight, we did bring Wendy Lee Stott here as a scientist to start a new project to address these questions. Is this one large population? Yes, but is it made up of a lot of smaller groups doing different things? So I would suggest you spend some time sharing your knowledge with Wendy Lee to guide her research. She's here to learn from you. The technology and looking at genetics has really changed a lot. Every year it's like changing a lot, and so there's opportunity to really dive deep. And again, I welcome Wendy, if you could just raise your hand, because that's what your research is based on, is all of your observations. Okay, awesome, thank you. And just getting back to talking about the whales that we captured and then died in 2019, we want to provide a summary as to what we learned with the group here today. So in 2018, 10 whales were captured and tagged, and those tags lasted quite some time. You saw Luke just shared with you some of the tags lasted up into 1 year. Oh, thanks. So the following year we were excited and we went out and we tagged 20 whales where we captured and tagged them. Unfortunately, some of these tags started to transmit from land. So we went and investigated, and sure enough, there were carcasses. So 3 carcasses were found and sampled. What we were able to do, though, because of the technology that we were using, we, in some cases, used tags on whales that were able to provide us a confirmation of mortality. So without the carcasses, we were able to confirm a total of 8 whales had died that were tagged. However, we suspect that additional whales had also died. We don't have evidence to confirm, but we suspect so, based on the transmission data received. So what did we do and how did we respond? So once we noticed this, and of course went to confirm those carcasses, we reached out immediately to our co-management partners to let them know. We also had to contact what's called the Animal Care Committee, so the people who provide us the licenses to do this research, to inform them of what had happened, as well as the officers, the DFO officers, to begin an investigation of us and our work. Shannon also reached out to all of our Inuvialuit team members. As you might have seen from some of the photos, we had a large team to inform them, but also ask for observations that they might have had or ideas as to how could something like this, happen. We reached out to our scientific community. So as you heard, there's people tagging beluga whales in Alaska and other places, other veterinary experts. We reached out to the expert community to talk about what had happened, to gather information. We had a lot of samples that we collected, so we started sending all of these samples out to all of the labs to look for everything, any clue that could tell us what was happening. As we started to receive data back, we launched into a community tour. We went to all the 6 communities in partnership with our co-management boards to share what had happened, but also gather information to help guide us and to understand, again, how could whales have died. So now we had information from different sectors and different knowledge, and we had gone back to the boards and asked, okay, what's a good approach now to bring all of this information together and try to understand what happened. So it was recommended that we would proceed, yeah, it's okay, you could check, that we proceed in bringing all of this information together in a big review meeting where we can bring together knowledge from, I'll say, ecologists like myself and others who are well-versed in looking at where whales go and had been involved in tagging programs. Also our veterinary experts who have more of a medical field understanding of whales and death. And also bringing information from our Inuvialuit knowledge and partners. So we proceeded with having a 3-day meeting in Yellowknife in March 2023 that was co-chaired by DFO and Game Council. So the meeting was hybrid to allow for our international experts to also participate. But at the meeting, we had Inuvialuit harvesters as well as some of our field participants and field crew and experts in all of these different subject areas to look at all of this data that was quite comprehensive in a 300-page document and to come to a conclusion together.
All right, so I'm going to talk about how we put all of this information together. So as Lisa underlined, we had a wealth of information, both from the field and from information that was gathered in the surrounding environment, and we have all of the harvest data, historical data available. We put all of this together to try and define, with the little pieces of the puzzles that we had, an image of what happened and what could have explained the unusual mortality that we had in the second year of the program, but not in the first year. So what we did, and that's where I guess the veterinary background comes a little bit into play, is to try and look at everything that could potentially kill multiple whales in a very short window of time, and using the information that was available to try and rank it from a most likely cause of death to least likely causes of death. This was going over 3 days, so I'm going to spare you some of the details and concentrate on what we concluded. But if you do have any questions, not hesitate, because we do have the information available. What we came up with is that basically, so the only common link between these deaths was that we did capture these whales 1 to 3 days before they died. So we are not blind that this is probably one of the triggering events. So with all of the other information available and having listed out some of the other possibilities, what we came down with is that we were able to narrow it down to a single likely cause of death, which is critical muscle fatigue and injury, which in my community we called exertion myopathy, and that's also something that's possible in humans. What that is, it's a condition that can be triggered by a muscle exertion, so you need muscle fatigue, but also the stress and fear associated with the situation. It's a well-known complication in wildlife. We mostly know about it from wildlife captures because that's when we have the animals in ends, that's when we tag them, and that's when we can follow them afterwards, but it can also follow natural causes. For example, I mean, and we know mostly about this condition from working with hoofed animals, so there's always like strict guidelines on how long you're going to chase these animals and in what conditions we're going to chase them, because people have encountered death in these situations. In cetaceans, we know that this condition can occur, and it's mostly been reported in cetaceans, so whales that strand on the beach. Because when they're stranded, they can breathe properly, they probably try and wriggle to get back in the water, there's stress and there's fear. But it has not been reported in belugas, despite all of the other historical capture programs that were going on. So the risk in our situation was considered to be pretty low, and the number of animals that we think died of it in the second year is very unusual and was very surprising because of this, especially when we consider that we were using the same protocol and we had literally the same capture team in 2018, so the year just before. So that brings the question, what was different in 2019 compared to 2018? And this is where we started digging a little bit deeper. The first thing that we did look is, did we make something different? Yes, we think that we use the same protocol, but we need to look at the numbers. So we looked at all the details for the methods. We came up with a score to look at the intensity of the chasing that we were doing to capture the whales. We looked at the duration and everything. We looked at if the whale behavior was different, including vocalization. And the answer to these analyses was that there was no difference between 2018 and 2019. So then we started looking at, and again, looking at the information that we have in other species that can have this condition, and that's starting to look at the local environmental conditions. So, and there we did find a major difference in that in 2019, for the animals that died, they seem to be swimming in warmer waters on the day of and up to 3 days after capture compared to the animals that lived. And that is a very important finding. As I mentioned a little bit earlier in my health talk, belugas are Arctic specialists. They are adapted to preserve their body heat. They have a really thick blubber layer for that. They are not well equipped to dissipate their heat. So if they exercise a lot, and then the way they dissipate their heat is to exchange, body heat with the water. If the water is warmer than what it should be, it could take them a lot more time before they actually bring their body temperature back to a normal temperature, and that can be detrimental for the body. And that could potentially have explained. It could be part of the answer of why we had more complication in year 2 compared to year 1. And here we can make maybe a link with so maybe we need to look at the weather forecast, such as for preparing meat, if you want to tag whales, because they need to have access to water that is of a temperature range that is okay for them. Then we also, in other species, another thing that we look at is sometimes some species or some individuals will be already more fragile and more prone to get this type of complication because of how they are at the moment you capture them. So not because of you, but just how like their health state, if you want, if they were, but not just health. It could be sometimes that younger, older animals can be more susceptible. We know that some species are more susceptible to get complication for a wealth of different factors. So we started to also look at what could have happened before, what the state the whales were in when they came in. And one of the most important findings that we found there, and that's using some new indicators to try and look at chronic stress, is that the whales that died had a more important indication of a chronic stress response than the whales that survived. And we think that this reflects their life in probably the weeks and months prior to capture. And as has been mentioned earlier by Manny, 2019 was a very weird year. There's been unusual mortality events in other species. Rafael can maybe speak to it in Alaska. There's been issues, and some of it was linked, I think, with malnutrition. There's been harmful algal blooms off the coast of Alaska. We're not used to see them. The whole year was pretty particular. There was also less sea ice cover, which potentially subjected the whales to maybe a harsher environment or a different environment than what they're used to. And then through the monitoring programs and the traditional knowledge that's available to us, there is indication that there has been throughout the years, so maybe more of an insidious change, a chronic change. There's been a gradual reduction in body condition through time, and there are indications that their diet is changing. So although these whales are adapting, when you put all of this together in a melting pot, what we think might have happened is that 2019 might have been a perfect melting pot for a storm, basically. And yes, capture might have triggered it, but something was different in the whales before, and something was different in the environment after, and all of this together probably led to all of this. So we can't control what the environment they are in, because there are changes outside of our magnitude, but they are changes that we all want to understand and better understand, and this might have been, like we say, the canary in the mine. One of the indications that they could be less resilient to something that we assume they have been resilient for, because there have been other programs where they have been chased longer and they did not have this rate of complication. The one thing that we can control is looking at the weather forecast, but yeah, that's I guess the conclusion of what we're looking in a nutshell for. So I don't know, Lisa, if you wanted to conclude. Shannon? Yeah?
I guess I just want to say, because I was the person who went to the communities when we first proposed this work, we talked about, we called it overexertion at the time, at being a risk. That could be a risk of death for these animals, but really we didn't expect something like this. We were more worried about a whale getting caught in a net or drowning. And so this was really surprising. And then what I heard from the Game Council was, we made the decision to do this work. We allocated this number of whales. We consider them to already be sacrificed from the population. And we made this decision with intention. And so that was really humbling for me and impactful, because I didn't expect this to happen. But the way that we brought together different people and different types of knowledge to do what we could to try to find out what happened was really important for us to have, a better understanding of what actually what happened. And I don't think we could have done that if we hadn't worked together from the beginning. And actually at the meeting that we had, the participants, which was pretty much our entire field crew, wanted to change the cover photo from a track of beluga movements to a picture of the field team, because we had this really unfortunate outcome, but we were also really we just saw Luke's talk of all the amazing things that we learned from the program. And so that was really a lesson for me to always really with intention think about what we're doing. We never tag animals for fun. We do it because we're trying to learn something, and when it's the only method available, we take that really seriously. And I know the boards did too, and they made the decision to move ahead with this program. And just want to thank everybody for making the work happen, to see all the amazing results from Luke, and for working through this problem together. If we didn't have the relationships that we have, this could have ended, and we might not be talking, and we wouldn't have been able to hear the information from Emily and Lisa about what happened here. So thank you. And we will take questions.
Well, thank you very much, Emily, Lisa, Shannon. Does anyone have any questions, comments? Okay, I'll go here first. It's okay.
Thank you. I don't want to put anybody in the spot, but I'm just curious, these dead whales, after they were tagged, were they ones that were net tagged or harpoon?
Only the net tagged, and so that's why we believe the capture was the factor that kind of linked them all together. Yeah, none of the harpoon tagged animals, we have no evidence to suggest that they were impacted. It's the stress of the capture.
Was the movement and dive data of the whales that died versus the whales that were alive, was that different? And was it different between 2018 and 2019?
So Luke probably has more details for that. My take home from his analysis, as we used them to interpret the data, was that there was no overall difference in behavior, in diving behaviors between the alive and dead whales. Nothing to say or make us think that they would have been like struggling a lot. Exertional myopathy in wildlife can take very different forms. And sometimes it can, one of the form usually happens between the first 1 to 3 days after capture. And it could be a sudden, cardiac arrest, or it could be that you don't have a lot of signs. So we think that maybe this is something that happened. But of course, like our access to carcass was very limited, and they were very decomposed by the time we got to them. And so we don't have a lot of data, but we do have that tag data. And at least, I guess, they did not seem to have struggled, and they seem to be behaving the same as the whale that died.
Hey, thanks. Raymond from Politic. I'm not going to again go into the question of what happened, but when we first heard of the program, when they were tagging whales, we were interested in how it was going to end up, because as well we had questions of where our whales were migrating through, coming from, going through, and overwintering. I guess my thought on this was that maybe it was a good thing that we didn't dive into it and start demanding, that we get some tagged whales, because I think we should treat it as a lesson, I guess, and use information we can to prevent any further casualties like that in that program. I can think of, a time in Cape Perry, Browns Harbor, on top of the cliffs there, we were able to observe whales migrating from Pierce Point to the east, straight outside Cape Perry. And I got to tell you that if I had a camera that we have now, we would have had a nice picture of hundreds of whales passing through. They were not bothering with politics, they're just going from one point to the other. And they had to be a lot of huge, large male whales. I wish I had, I still can picture in my mind, we had the vantage point we had on top of the cliff. We could see them for miles, that one line. And it makes me think that maybe to enhance observation, we should post somebody out there from as soon as the ice opens to when it's done, just to capture the amount of whales going through. Thank you.
Hello. My name is Clara. I'm from Inuvik. Imagine getting a sliver and not caring for it. It goes to blood poisoning, then death. So that's probably what happened to the whales. Because at one time we saw one of the pictures of where the whale had a lot of pus around the tag.
No, so the possibility that the tag got infected is one of the possibility that we assessed in the assessment. So on the tagged carcasses that washed up, this is one of the tissue that we have been able to look at. And then we also did a critical review of the information out there on potential complications associated with tag. The tissue that we were able to observe from the tag only showed very minimal and expected inflammatory cells, such as after a trauma, and no sign of infection. And there is one case of in the cases where there has been complications associated with the tags themselves in the literature, it's usually cases that take a lot of time because you need infection to have the time to develop around the tag and before it goes into the system. And there was also strict disinfection methods in the field to reduce above what has been done in previous tagging programs with no problems too. So altogether, when we put that, I mean, we can't rule out that maybe there could have been infection in one well, but with everything that we did and the information that we have on that type of procedure and how we're careful not to go too deep into the blubber, we don't think that it would have happened to so many different wells over the course of the program. So this is where looking at what can a common cause for multiple individuals in a short period of time. This was and we looked into all of the details and tried to and use all available information, and that was considered as a least likely cause compared to a complication with capture in our case. So, but we did look very deeply into that one too. Thank you.
You could hear me? Okay. If you didn't tag them, would that be alive?
Yes. If we you say if we didn't tag them, so I just want to make a distinction here between capturing and tagging. As we underlined, all of the harpoon tagged individuals survived. So we do think that, the condition that we think have happened, it's associated with muscle fatigue, stress, and fear, and that's mostly associated with a life capture. So it's not the fact that we tagged, it's the fact that they were life captured, and it's not just the fact that they were life captured, it's the fact that they were life captured and it was too warm for them after, and they seem to be fragilized before. So there has been multiple points that need to be taken into consideration. It all plays into risk analysis for these type of methods, and all of the work that we've been doing collectively as a team is going to inform animal care committees that oversight these type of procedures, because this changes the balance of risk. At first, we didn't have any information or indication that this was a potential complication in the program, but with all of the information that we gathered and the red flags that we can now see looking at temperature, especially for belugas, this is going to help inform and make sure that future programs are safer. And again, like Shannon highlighted, this is an invasive procedure. We all agree on this. This is stressful for the animals. It is only done if it is the only way that the information can be gathered, and it's deemed information that's important enough for the population. And it goes through a very thorough process. And all of the information that we were able to gather collectively is going to help for the future.
I'm happy to, I say thank you for all the information that you find. But when you do things like this, think of if it's happening to you, what would you do? What kind of help would you get? Because you're doing it to another animal.
Absolutely, and I think I can talk for a lot of people on this team that this thought went through our head. I think we all went through very difficult personal, professional moments following the discovery of the death of these whales. I think it all started, it was a long healing process with huge help from the meeting and getting together and finding answers. But there's always going to be a little scar because this was not the intended outcome for the program. Yeah.
Thank you, Clara, for your. Do we have any? Oh, that's right. Okay, Jim, we'll go with Jim first. That's okay.
Yeah, thank you, yeah. It's on? Yeah, I'm just wondering, like, to me, I think that's a lot of good information you're getting from the tagging and the whaling and that, and then just wondering, like, with our neighbors, the U.S. Fish and Game on that, when they're doing tagging and stuff like that, and when you guys did your work during the 2019, you guys were more efficient and stuff, and there was like, you had in the whale for only 18, 19 minutes. I'm wondering how long was our neighbors handling their whales in that, because with the stress level, I don't know about it would kill a whale or anything like that, because our neighbors in the U.S. side, we hear them herding whales for 7, 8, 10, 12, 14 hours long sometimes to get them back into a bay from out in the ocean and that, and you'd think that'd be a lot more stress on that whale being herded for hours and hours, not being handled for 20 minutes and that and stuff like that. So just wanted to bring that up because I was going to bring that part up because you didn't really show how you guys improved from 2018 to 2019 by a couple of minutes and stuff like that. It was more efficient. Thank you.
It's a good point in the, I mean, of course, we had to summarize a lot of the information, but there's a nice graph that has been made into the very big report document, the summarizing all, where you put the total times for procedures on the whales from this program compared to what is available as information for the other programs, and we're much lower altogether, and 2019 being even more efficient than 2018. And I think Rafael has a comment there too, maybe from to speak on behalf of the American side.
No, I wanted to respond to the driving time. So yes, it's a long exhaustion, but they're driving slow. You know, because they're trying to get them into lagoons. Now, I have not been, part of the driving, but what I'm understanding is the way they're doing it's a slow drive, because normally we wait about, 10 hours before they're able to bring them into the lagoon. We have looked at muscle tissue, actually, on some of the whales just that were driven like that. And we very, very occasionally actually have this type of lesion, what she's talking about, that exertional myopathy. So we know that the animals are driven slow enough, just like when you're driving deer, when you're driving reindeer to get them into the corral, you just take a slow pace. And I think the, what's being shared here is really, it's the combination of really warm waters, right? I mean, I remember I was that year on Cross Island, and it was glassy water, and we talked about you could go and nearly, swim in it. I mean, it was amazingly warm, the water, right? So I think we're really talking about unusual warm temperatures and maybe not being able to, get the worms off them, right? Dropping them off the back. So just wanted to say that. Thank you.
Rachel?
Thanks. That actually kind of leads into my question quite well, too. I wanted to bring it back to the environmental factors, because you did mention that the warming temperatures could have been a factor in some of this mortality. And climate change is here, we know that, and over time, temperatures are only going to get warmer. So I'm just curious on how that will impact future programs or guide any of that consideration for future tagging. Like, are you kind of looking into or testing that temperature before you go out, or how is that going to impact long-term as well when these temperatures aren't really so manageable as well?
I guess I'll start, and I just want to be clear that we're not talking about a new capture tagging program in the ISR. At this time, we're not talking about that, but there are tagging programs still happening elsewhere in Alaska and in the Eastern Canadian Arctic, and so what we've learned from our experience could apply there. And so there's a number of things we would do, and Emily mentioned it. We would definitely look at the local temperatures at the time of capture. We would probably look at what was happening in the winter if we had some of that information, and if there was a decision to have a tagging program, we would probably do something like introduce, they call it administrative controls. So we would say have a much lower number of animals that we would tag,
And we might, we always monitor once we tag the animal and release it to see where it's moving and how it's moving and how it's doing. But, maybe we would space that out more. There's different ways to address that. And then Lisa mentioned that there was a toad tag that pops up when it's at a constant depth for a time that you set, which is usually 24 hours, and that usually signals death, because why would a whale be at the bottom for 24 hours? So that's another thing that we would recommend and add, one of those tags that will tell you that an animal has died, because otherwise, if it doesn't wash up on shore, we don't know, really. We just know that a tag stopped working. So we were really lucky that we had some of the technology in place to learn more about what happened. But definitely, climate change is something that we have to, like Emily said, it's changed the risk profile. We've tagged belugas in the Canadian Arctic for 30 years. There's been a few tags that have stopped transmitting over a short period of time like this. We didn't know what happened. People just thought the tag stopped working. But overall, things have gone really well. And so now we need to think about climate change when we're running programs like this.
So I guess my question is, the Mackenzie River is very warm. Tuck often has the warmest beach in all of Canada. So, every other tagging program across the Arctic probably took place in 4-degree water, and you guys were 4 times that. Was any consideration in the water temp put forth before you went out and tagged, I guess? Like, did you think, oh, we're going to be tagging in 20-degree water, whereas everyone else tagged in 4-degree water?
We weren't thinking like that at the time, because there's been tagging programs in the ISR before, in the early 90s, the mid 2000s. So that is something that we would communicate to other scientists running programs. Keep that in mind, and that's something that we are talking about with our scientists we work with in the Eastern Arctic. We had in 2018, we didn't have this problem, but we had ice around us for a lot of the time that we were doing the work, so it was very different.
I think people are getting ready for need a break, but we'll take one more question, or Manny, and then we have a question here. But then
Really short.
Yeah.
Good. Just trying to wake up everybody here. So. Good afternoon. Just to let you guys know, whales, when people do sink them, and they sink, and then a couple of days later, or 3 days, they float again. I just want to let you know. They don't stay down. Thank you.
Good afternoon. Lenny Imacho Kumtak. My question is going to be, when Alaska put the backpack on, is that the same system that they use in the Canadian side? Because I got a whale one time with everything on it except the backpack. The cable was there, the bolts, everything was there except the backpack. Do they use the same system?
They do. Would you like to answer that? Yeah, they use the same style tags, and they call them spider tags. So there's 3 kind of wires on either side, and the one that you got, because it was earlier, it might have had 2 wires, or maybe it had the saddle, but it's the same attachment method.
Yeah, I sent it over to Lois Harwood. She wanted it, so I put it in a box and sent it off to her. Thanks.
Yeah.
Yeah, I just want to ask, when they did the tagging, like in Alaska, did they use the netting too? And haven't heard if they had any fatalities too, like we did in the Beaufort. Thank you.
Eileen, you're asking if they use netting, if they net the whales? Yeah, so I'll start, and Raf, you can correct me, but I think, Raphael talked about the bringing whales in, herding them in, and then tagging them. And I know that sometimes Robert Saddam had talked about using a set net. So we go out and encircle the whales, which we had on the front here. But you can also just set a net and you can bring the whales into the net and capture them that way. So I think that's what you're doing. Yeah.
Hello. So, yeah, both techniques. It's either that you're driving them in, and I got to make that clear. So, in general, the drive hunt is for the community to hunt whales, right? So the tagging always happens afterwards when we are done. So when the community has gotten their whales and they moved off, that's usually when the, the side, like John said, the people who are involved with the tagging mostly, that's when they stick around and see if there's still belugas in the lagoon, right? And then, use multiple of the, the inflatable boats to kind of, herd them towards the beach and then, capture them. But yes, otherwise it's the same technique. But actually, I think you guys have been much more successful in tagging, than actually out of Point Lay.
Curiosity here, Rafaela, how much time is there between the drive hunt and the tagging? And do you think that this is like, and how much time is there when you can, how much, what's the interval between a potential drive and the muscle injury that you're able to document in these wells?
I think it's really complicated to answer. All I know is that, it sometimes takes them, can take them up to 8 or 9 hours to bring the whales down from further, right? Because they have to use the access points, right, into the lagoons, right? And when I was saying that we saw on histology, right, that's sort of looking under the microscope, right, nothing that I saw on the whale, with my eyes, we saw some evidence in small areas of muscles, exertional myopathy, right, and that's what you would call it. But otherwise, there was no evidence that the, whales were particularly stressed, and I think you can't drive whales if you're not calm with the boats and slow, and you take a really slow pace, because they're just going to break up. At least, I'm sharing with you what hunters have shared with me, right? I've not been out on a boat, but this is what I hear, how somebody, the boats don't cooperate well. That's how you have them splitting up and you're not able to get them back. So I really think the driving is not as stressful for them. I think they're just, kind of being guided. When the chase in the lagoon probably happens, right? After it, yeah, definitely there is probably some flight behavior. Does that answer it?
Okay. This is a really, really important discussion, and I want to make sure that everyone has had a chance to ask their question and make their comment. So just before we go to the break, any final questions? Okay. Jimmy, did you put your hand up there? Oh, you were quick. Okay.
Hello. I think maybe on a positive note too, there was a whale harvested with one of the hunter harpoon deployed tags. The ones that we developed was found inside one of the harvested whales, and there was no infection, no pus, no nothing around it, and it healed very well.
Very good to know. Okay then, so I think with that, everyone needs a break. I noticed some food has been brought in, so let's take a 15-minute break here. You take the opportunity to look at some posters, but we're also going to have a poster session later. So thank you very much to the presenters, to everyone here. Please come back for 3:45. All right, could I get everyone's attention, please? You don't have to go back to your seats or anything if you're still getting food, if you're looking at posters. we are going to go into the last session of the day, which is a breakout group session. And really what we want to talk about here is bridging Inuvialuit knowledge and science. And we're going to do so under 6 different topics. So we're going to have a breakout group for country foods, discussing things like health benefits, safe preparation methods, culture and values. That group is going to meet in the curling club. The second group is about the health of belugas, talking about things like disease, parasites, and body condition. That group is going to meet behind the curtain here on the stage. The third group, contaminants and plastic litter. We are going to meet right here in this front corner of the room. I say we because I'm going to be in that group. We're going to talk about the chemical contaminants that are present at low levels that are introduced by human activities, primarily in the south. Group 4 on population structure. You're going to be back there by Marianne, where the exit door is. Talking about how many whales are there, how are the whales organized, size, sex, age, etc. Group 5, movement and migration by the water cooler, by that pillar back there. You guys are going to be closest to the food. It's a lucky group. You're going to talk about habitat hotspots, big picture movement from summer to wintering areas. Group 6, behavior and habitat use. You're going to meet by the drone poster, which is over there with Kim's waving her arms. You're going to be talking about what do belugas do and where do they do it. A local scale, could be a large scale too, I presume. But let's remember the theme of this is bridging Inuvialuit knowledge and science. We want to make sure that these groups have a good balance of scientists and Inuvialuit knowledge holders, so we have that balance in each of the groups. Now, we have assigned, so there's some facilitator questions here. Each group will have a facilitator and a note taker, and these are just some leading questions. I'm not going to read them out in the interest of time. These are meant to help inspire some conversation. Okay, now assignments. I don't know if everyone can read this, but we've sort of made suggestions for where the science participants could go. We've got named a discussion facilitator and a note taker. Can everyone read that, or do I need to read those out? We're good? Okay. All right, so with that, I think those are our instructions, those are our assignments. We can each make our way to our station. I'll put that back in case you've forgotten already. Okay, and we'll get that conversation on the go. Now, it is fairly late in the day. We got a little bit behind schedule there, and there's no problem with that. We had fantastic conversations, but this is going to be a little bit shorter. We want to get everyone back in here for 4:30. I'm sure Jen will be walking around herding everyone back in here at 4:30. Okay, so off we go. Okay. I hope everyone had a great discussion in their breakout group. I know we had a really great discussion here about contaminants at the front table. It's now 4:45, and I think Jen's going to give her wrap-up comments and an update perhaps on timing of the dinner and things. I just want to say thank you one last time for having me be your chair today. It was a pleasure, and I really enjoyed it. So thank you very much. Jen?
Thanks, Jason. You made my job really easy today. I don't know about you, but my brain's full. It was a lot today. It was a lot, and I really appreciate all the work of the researchers, those who presented, those with their posters, all the work that goes behind all that work is immense. Throughout the day, I was listening, not just the presentations, but the conversation across the room. Two things kept coming back to me throughout the day. Firstly, it's evident the passion and dedication of the researchers working up here in the ISR on these issues. And it takes a certain breed of researcher to work up here. Another was something that Sonia said about research being a form of reconciliation, a form of healing between groups. So there was recognition that there's been research and policy work done up here in the past that has not been respectful and even harmful. When Chloe from GWT Public Health acknowledged that her department in the past have not provided messaging that has been helpful to the Nunavut and that they are going to be doing better. It's noted that research is important, but researchers need to be careful to not disrupt the community's confidence in the healthiness of country food. And once again, driving home the message of the importance of belugas and other wildlife's role in the lives of a Nunavut. Brian reminded us that country foods are not a plan B, right? They're not a luxury. They're a first choice. And that traditional foods are a key for a healthy diet up here. The land, in this case, the ocean, heals you. And the importance of country foods is not just for its nutritional benefits, but also for its physical and mental health, well-being, and pride. Because protecting the land is intimately tied to maintaining a traditional diet. Our afternoon session focused on the population estimate and the tagging program, and now we have a much more detailed and comprehensive story of a life of a male beluga from Hendrickson, at least. that's one little cohort we have a better picture of. From both researchers and community members, there's an appreciation for the diversity of beluga activity. But with more information, it just led to more questions. So, I think, we have to see this as an iterative process. When it keeps coming back to us, more questions will arise from that. And Luke, just see it as job security. The work was unfortunately overshadowed by the mortality incident in 2019. And the gravity of that, despite all the good work and intentions of researchers, Nunavut boards, and the field crew, it drove home that research has its risks, and the constant weighing of the potential impact of handling and disturbing wildlife needs to be placed against the need to know more to better inform management decisions. And it's not an easy process to navigate. But as Shannon noted, it would not have been possible to come to understanding what happened in 2019 if it weren't for the already existing strong bond between researchers and the Nunavut. A relationship, in this case, forged through co-development of the tagging program. A process that saw Nunavut involved in even driving much of the work. So both researchers and Nunavut need to be commended for taking ownership for their part of the event, intentional or not, and for coming together instead of against one another in trying to figure out what happened. And I believe there was a form of healing that came from working together through a difficult journey to come to a greater understanding and to move through and pass it. What came out was a new knowledge that could help inform future work and the work elsewhere to hopefully prevent future research-related mortalities of this nature. So you should all give yourselves a hand for that you all came together to address this issue. It was challenging, and I was there at the time. I was a game council staff, and it was something that I didn't know if we'd be able to actually work through it, honestly. So that was my take from the day. So thanks. So, we had some food that already arrived. The feast itself will begin. We kind of moved it back to 6 o'clock, just to give people a chance to run back to their hotels or home, and then come back here. And so we'll begin the feast at 6, And we'll see that we'll also have the video for the Politics drone project. And we'll have a chance for you guys to talk to the presenters for the posters. All right? I'll see you all in an hour or so. Thank you.